problem for conducting the trials. Is there any window in RSL trials? The DFF is 120 to 130 days, but the entries were receiving 145 days DFF. So that will be creating a problem to conduct a trial. As well as we have receiving from last 3 years seed by 10th of July. So that will go up to the December 15,
Listen, you are telling a specific trial for your location, that is not possible.
No, location.
130 days duration, all they will treat and send for you.
So across the North India,
no, that is wrong suggestion.
No.
But please tell you tell.
Yeah. What Dr. Prakash, you are telling, actually we request all the cooperatives to send the seed in time. What is the problem in RSL? We have only few entries.
So next year we can drop the trial.
No. Your concern. No, Dr. Prakash, I understand your concern. We are trying to send it as early as possible. This time I request all the cooperatives to send seed as quick as possible. Otherwise, it is not possible to include your trial. Include your entry. That will be taken care. Okay. Yes, and regarding second I am coming. Regarding the duration of the entries. If it is late duration, then those are dropped actually. Those are not promoted. That's why if it is in first year, you are not getting sufficient time, and then you report it. It will not be as per the monitoring. Those entries may not be considered for the next year. Okay? Yeah.
Good. Very good. People send it in right time. Then they will send it. If they are delaying, we will be telling them. Whose fault is it? We don't know now. Yeah, please.
In a concern, not only in rain-fed trials but also in irrigated trials. When a window is set, that before this date you have to send the seeds, and in advance you have to tell these much number of nominations you are making. Kindly stick to the schedule, because if seeds are received late, then automatically the trials dispatch will also happen late, and finally report preparation will also happen very late. And another concern in all the trials is that many times data from that center is coming very late. We have set a window. What is the window we have set? I think December last week or January first week. By that window you have to send the data. If it is received beyond, it will become impossible to analyze and then put the report in time. And this time we faced a very tough time, because from many centers the data was not received, and then we had to contact them, and then only data was sent. So my humble request that please timely dispatch your seeds, timely conduct the trials, and if it is getting delayed, get back to us that because of this reason it has got delayed. What is the point in conducting the trial?
Yes, sir. Already we have consumed 1 hour for breeding, and there are another 5 time is up. How to manage? So quickly or
just one point. Dr. Das, or ABT need dot trial. Whether you promoting any entries from last year trial, I did not see any in the slide.
Last year trial actually that one entry is I think it is in second year trial, but it has to be confirmed in with EDS trial. That's why.
Is ABT need dot trial?
One entry. Nil trial I am talking. Nil trial into the second year, it is not confirmed along with the ABT 2 and 1 EDS. That's why I think no entry is there. Only one entry, it is there.
No, it's not related to the DS trial. It's ABT need dot with medium duration entries.
Yeah, actually.
One, please talk. No time.
Here you can see Nil trial there is no entry.
No, not for this one. ABT need dot.
Yeah, ABT.
Again, you are from the same institute.
Okay.
It will take time.
Yeah. We will discuss.
We will discuss one.
Okay, thank you.
So I think we cannot be more. Already we have taken part in the whole time. How we can allow more questions? Thank you, Dr. Das. Any promotion criteria you are facing difficulty? Yeah.
Actually, just I want to emphasize. Initially, it was CD. Initially, we were promoting on the basis of 5% superiority. Later on, it come CD value on 5% with CD value. Then, from last year, the committee has decided to 10%. Still, we are facing difficulty because very few entries have been promoted. That's why initially we are reiterating, if it comes to the earlier the condition of 5% promotion, it could be better. Otherwise,
you do one thing. I suggest this. You make both, either of this.
yeah,
It is if CD is okay, is not okay, then both numerical superiority.
yeah.
5%. Isn't it okay?
So,
Yeah.
I've been discussing with you.
Yeah, it is great. No? This is the group. If both simultaneously, so we are not pursuing either of these.
Numerical
Like
means
it is
how much percentage?
5%. Like irrigated.
Yeah. The
If that
irrigated
is-
is 5% CD only.
Now, even
No. 5% numerical superiority.
5% numerical we can take up. It is need not in turning area. CD only. Yeah.
Acha, very good.
CD only.
You want to go to the older country.
Yes, sir. We want to
So
go to
House,
order.
please suggest. This is very important. So very quickly we have to decide it.
said that.
Yeah, please tell.
Whether 5% will be make a significant difference in terms of yield under drought conditions. For example, if your best check yields say about 2000 kgs, how much is 5% of that?
Sir, okay, I understand your concern, particularly in nail drought and nail submergence, we will be sticking to the CD value. That is true. But in case of other trial, particularly EDS and semi-deep heat, the 5% earlier what we are considering, that I think it is a kind of acceptable,
In case
reasonable.
of stress-prone environments, actually, replication maintenance CD is very difficult.
Very difficult.
That's why they are not getting superior.
Actually, it is very difficult to conduct trial. 3 trials, there is no promotion.
CV is going up to 40%.
Yeah.
But now CD is going up to
40%.
That means it is difficult. They are unable to manage. So if under such error, if we will be targeting CD value, then it is promotion they may not get. We are allowing so much of error. From the real world, where to get?
Sir, you
So it is
see,
better to go for
semi-deep
numerical
deep,
superior.
there is no promotion. SGW, no promotion. RSL, RSL also very few promotion. How to constitute trial? All the trials will be stopped.
I invite any negative comment about numerical superiority. We should adopt or no?
Biren Singh sir, can you tell your suggestion?
Not on the computer and so once you visit the trial, अभी monitoring tool जो है उसमें आपने कहीं नहीं लिखा है कि this increase will be dropped. There is a severe infestation lodging there after past inspect and nothing, and then we promote the entry. When the ABT, this is happening. So the monitoring team has to be very
Meticulous.
critical, and any entry which is lodging even in the ABT stage is being promoted. Even the whole book you see, they have taken the primary person. That has no relationship with the field. So we have to take the phenotypic acceptability of a breeder at a particular location into the criteria for promotion.
Yeah.
So I do not know the plant breeder, they are all arguing on the laptop breeder. We are not selecting based on the field. So they have to select in the field. Kindly visit them and then you see which is the better entry as compared to the check.
Okay, regarding sir 5% promotion, your suggestion.
Let the entry tell whether it's better than the local check or the national or whatever check. That significance is not met. If it is such a good particular disease.
Okay. Okay, sir. Yeah.
So, what did you get?
Sir actually he is giving his consent regarding 5%. But he is telling to be critical regarding the entry, whether the entry is performing well or not. In that case, the nominating the monitoring tool will decide whether that entry would be
My concern
promoted
is
or not.
that even though your intentions are noble, but it will become subjective. Because for me, something that is phenotypic score of 9 may be 7 on part of my partner who is coming for monitoring. It.
That's why the environments are very variable. That's why we are not able to make progress. It's not the irrigation problem.
Let us try to continue in the same way. We are unable to get a consensus here.
No, sir. Sir is telling it is 5% is okay.
5% CD or 5%
Not CD.
numerical?
5% numerical
So
superiority.
why so many discussions? Let
No,
us have-
sir's concern is different.
So why other things we are taking care?
Oh yeah, we'll take care. Sir, your other concern will take care. Definitely.
I have many concerns, so let me tell another thing.
नहीं टाइम नहीं है सर.
टाइम बहुत है, आप सुनने और डिसीजन लेने की जरूरत
Sir, we are eating into the next session, sir.
नहीं, हम 1 मिनट
तो सेपरेटली मीटिंग करेंगे. सेपरेटली यू अरेंज वन मीटिंग ऑनलाइन या समथिंग. ओके.
एक ऐसा है, डॉक्टर बाबू इज नॉट हियर, अनिल बाबू. लास्ट ईयर उन्होंने एक कमेंट किया था कि ICAR-IIRR does not conduct any trial. All the AVT and IVT trials should be conducted by the scientists of the ICAR-IIRR. हमने पता किया तो ये पता लगा कि जब वो डायरेक्टर हुए उस समय से बंद हो गया. And last year he made a comment in the IIR meeting कि IIR को भी ट्रायल करना चाहिए. तो जब आप ट्रायल करेंगे तब ये 5% सीवी का पता लगेगा कि what should be the criteria. The plant or variety is fixed in the field. Which one is the better? So I will request Dr. Sundaram कि जितने भी ट्रायल हो रहे हैं. Earlier I used to go with Dr. P. Ram and see the trial at Vikiset farm. अभी मैं आता हूँ, मैं कहीं देखने नहीं जाता.
Sir, 1 minute, sir. We don't conduct the trials in all the typical replications, but each and every season, it's a wrong thing what you are telling, sir. And I make it a point, just listen to me, sir. I make it a point to see that seed is multiplied at least in 1 or 2 replications, and each and every entry is monitored. And
It's not the notification.
Dr. Das, it is
It
clear.
is evaluation.
Yeah, yeah.
sir, it doesn't make sense to conduct the trials when already we have got another institute,
Dr.
which is
Sundaram,
yes, sir.
Whether you got your criteria or not now, I want to invite the next person.
Yeah, yeah.
You
Now
got
it is
your
clear
point?
that in the house that the 5% numerical superiority will be taken as it was taken earlier for the promotion of rain-fed entries, particularly. Leaving aside the trade verification trials, that nil of drought and submergence.
Sir,
Other trial will be taken on 5% numerical basis.
Yeah,
Thank you.
okay.
We can consider first CD. If no entries are going on, then we can go to that
Yeah,
5%.
yeah,
Okay.
that is the condition.
That is the condition.
Yeah, both, that's
Okay.
why I told
First you
both
must
see.
go by CD. If the trial is not having any promotions and you are not able to progress, then only you can.
apply the
Okay.
second.
Okay, fine. Fine.
Both that means exist.
Okay. Thank you.
So thank you, Dr. Das. A lot of discussion about this. So now I request Dr. Mohinder Kumar for agronomy. Quickly repeat your charges are less. Then 40 now. Please finish 10:45. Just important changes, highlight what you have done, please say those. Routine thing is not to be reduced.
Sir, very good morning to everyone. Respected Chairman, Dr. Pradhanji, co-chairs, Dr. Aram Sundaram, Dr. Vaig, and rapporteurs, my dear colleagues. Only 5 minutes, sir. Hi. Sir, we have thoroughly discussed, sir, during not only in our presentation by our cooperators, and also during 26 and 26, and we have thoroughly discussed, and we have finalized our technical program. I think in this agronomy evaluation trials, we don't have much say in this one, because these are the lines promoted by breeders at AB22 level. Those things will be evaluating for under optimum as well as suboptimum level. Only my concern here is timely receiving of the materials. I think already Sai Prasad has told that any AVT2 entry has to be tested. It has to come to the head agronomy as early as May 15th. So after that, any entry comes, we are not going to include in the AVT2 trials. It was, I think, by May 16th or May 20th, I will be sending this, what are the entries we have received for this evaluation under AVT2 trial. Because our own agronomy people, they told that we cannot evaluate if some entry comes at June 15th, July 15th, and we cannot see the potential of that entry.
Sir, when somebody sends it extremely late, communicate it back to them also.
We'll send it, sir. We'll send it on 20th itself. We will send it that these are the entries we have received and these are the entries are going to be tested. And we need sufficient quantity of AVT2 seed based on the number of locations we are testing, and also pure seed. Some of the AVT2 seeds are also mixtures. That is very unfortunate. I am very sorry to tell these things. Please see that. I think we will get a good quantity seed of AVT2 at right time, and so that it will be distributed. Even if something is monitored by NRRA Cuttack, 2-3 trials are there. For them also, the AVT2 entries should go to the agronomist. So with regard to the, I think they are the locations which they have already tested it earlier. So that's only I have mentioned it. So there may be some more changes in the, and if you can send it that local checks also, checks for these same entries, that will be very useful for us to save the time. And otherwise our breeding section will provide whatever checks are available. Under resource conservation technologies, we have already listed this. I think as per the suggestion, water management has to be focused, and now we are going for total water management under direct seeded rice, and we wanted to see that IoT devices can help us in computing the water input and other things, water savings. As suggested, we are focusing on DSR methods, and we will be specially giving a protocol for DSR system. I think I will be taking help of IRRI and others also before sending the protocol. So this protocol has to be strictly followed wherever this DSR experiments are conducted. We have both sets of DSR dry and DSR wet system. We have 3 trials. I think still we are promoting under weed management. That long-term weed survey will continue. And weed survey we wanted to see further. IPM trial will be conducted not only with agronomy, entomology and pathology. This is, I think, 2 trials, sir. This evaluation of sorghum hybrids we are, I think, discontinuing because we don't have any entries that hybrid suitable for Rabi season. Conservation agriculture, I think we need to focus more on. I think our DG is also emphasizing how we can do the conservation agriculture and make it very profitable. Our interdisciplinary trials will continue, sir. We wanted to see the yield maximizations, maybe little bit changes in the treatment, sir. But I think we are going to conduct these yield maximization trial and natural farming by our soil science head and nano fertilizer. I think they are going to study for 1 more year. Drone, I think many people are, I think, wanted to test this drone-based herbicide application. Even we wanted to see that drones can be used for seeding. We have a technology by PJTU. They can do it line sowing of the wet seeded condition. They already got patent for that one. So those things we wanted to test it. So these are some of the new initiatives. I think we wanted to go for a little bit emphasis on climate resilient technologies by agronomists. So we wanted to use some microbes which can also take up the methane, and it can also improve the productivity of the rice. So we wanted to use some clean rice products, and one of our cooperators told that they will also supply biochar. We wanted to use it to biochar application, and as I suggested, IoT based devices and DSR collaboration with physiology. They are going with a very big way to conduct a DSR trial. We wanted to evaluate those entries in our agronomy. So I would like to thank you, sir. Thank you. This is about our technical program for the year.
Thank you, Dr. Mahendra.
2025.
Actually, very good trials you have suggested. I hope it should be accepted. If anybody is having no to this, please, Dr. Dev, please.
Specific query we are having, that concern what Mahendra Kumar has raised, that timely sending of seed, you should maintain that one, and you should not entertain all those seeds which are received after 15th or so.
Yes,
You can and purity of seed, that is also a concern. So all partners should consider all those things. And any further, any query, any suggestion on this presentation, you can tell.
Against new trials, if you have anything to say, say no, or else these are good.
Sir, it was thoroughly discussed, sir. It was also suggested by our
Already one trial.
Subbarao Garu and then Sesai.
Okay, then yeah, I don't see responsibility.
Understand that DSR trials, breeding trials is being conducted by agronomy. They are also included as part of your trials, and they'll continue as it was done in the best possible way last year.
Yes, sir. We will be giving a protocol also for that, wet DSR and dry DSR. That will be also taken by the breeders, sir. Same.
But data will be collected not by breeders but by
agronomists will be conducted.
Agronomic trials data will be collected by agronomists.
Yes, sir. Yes, sir.
Yes, by breeders.
But even management also, our agronomists are doing that, especially for DSR. Sir. DSR is very difficult to establish, even population also. It is very difficult. So agronomists will be associated with the breeder trial also. Thank you, sir. Thank you for not having any queries. Thank you very much.
Very good.
Okay, very good. So next, I invite Dr. L.B.B. Prasad Babu for soil science presentation.
Thank you very much, sir. Respected Chair, Dr. S.K. Pradhan, ADG. Respected co-chairs, Dr. Sundaram and Dr. Beg, deputies, and all the participants. A very warm good morning to everybody. I will be presenting the finalized technical program of soil science, which was discussed for the last 2 days. First day in the crop production, among the crop production section, and yesterday among all the participants of all the disciplines. Sir, we are conducting 9 trials, sir. First one is the long-term soil fertility management and rice-based cropping system. This is the 36th year. Here, there was a suggestion that study of the microbial biodiversity of the selected treatments is to be taken up. And it is being conducted at 3 locations, sir. These are the set of treatments. Next one is the next 2 are the management of sodic as well as acid soils, sir. These will continue. It was recommended that the present set of treatments among these 2 trials are to be continued for the 1 more year, and then we should come up with a fresh set of treatments among these 2. This is the treatment set of sodic soils which are currently being taken up. This is that of the acid soils. And then 4th trial is the residue management and rice-based cropping system, sir. This experiment will be continued for 1 more year and it will wound up, and it will be taken up at 10 locations as far as the Indians received till date. And the 5th one is nano fertilizers for increasing nutrient use efficiency, yield and economic returns in transplanted rice. This was agreed to be continued for 1 more year, and it was suggested that more focus should be on nutrient balance and soil health impact in the long run in foliar spray nutrients. The Indians have come for from 11 centers so far. 6th one is yield maximization of rice in different zones. It is a collaborative trial, like in the previous trial, collaborative trial with agronomy. This will be continued with the same set of treatments for another 2 years. And it can also be, there is a suggestion that it can also be studied in DSR environment wherever feasible. And Indians for 13 locations have come. 7th one is enhancing productivity of organic rice cultivation and natural farming, sir. Here, there was a consensus that a separate inorganic treatment of recommended dose of fertilizers is to be included just to see the yield decline in complete natural farming treatment. And then 8th one is zinc response of biofortified rice genotypes to assess an agronomic biofortification potential. This is being coordinated by our counterpart CRRI. This is the 2nd year of trial is over. It will be continued for 1 more year. And then it was suggested that to identify the variety with high zinc content as well as yield potential and farmers' acceptability. This is the last trial, sir. It was recommended. It was started only last year. As a prelude, we have conducted a survey. And then this year we'll go full-fledged, and it was approved for 1 more year. And carbon dynamics in virgin soil vis-a-vis rice cultivated soils is to be studied. This will be taken up in 10 centers. This is the treatment setup. Thank you, sir.
Thank you, Dr. Prasad Babu. Very good presentation. You have already discussed with the soil science. Yes, sir. Yes. So, any other persons want to comment about this? If no comment, then we can.
Or if you have any concern on this trial,
Yeah, Dr. Sundaram.
The experiment on microbial diversity, are you just going with traditional methods wherein soil sample will be collected, microbes will be subcultured, or are you also going for metagenomic analysis?
We will take a metagenomics.
Yeah, that will, whatever traditional approach you do it, but apply metagenomics
Yes.
also. It will not take that much resource. It will give very good information.
That word is missing here. Yesterday in proceedings we kept it.
This was suggested by our Honorable DG sir
Yes,
also.
we will include it, sir.
Okay, very good. No question. It's clear to all, agreeable. Thank you.
Thank you, sir.
So next I invite plant physiology, Dr. Raghuvir Rao.
Yes, sir.
Yeah, we are on.
Good morning, sir. Dr. Pradhan sir, Dr. Sundaram sir, Dr. Beg sir, and rapporteurs and my dear colleagues. Since last 2 days, we have been discussing critically about all the technical programs, and we have finalized those. But however, for the larger audience, we want to share with you what plant physiology has to offer in this technical program. Sir, we have details of to be conducted. The 6 trials are there, sir, in plant physiology. The first one, silicon trial, was conducted for 5 years, and we have come to a conclusion. A technical bulletin will be released on that, and we have dropped that silicon, sir. In its place, we have direct cereal rice, which will be taken up with physiological implications, will be studied for this year onwards, sir, in collaboration with soil science and agronomy. In screening of elite germplasm for phenotyping for drought tolerance will be studied, sir, as usual, and this trial also will be conducting in case of irrigated and drought conditions. Third trial, sir, screening of high temperature stress for rice genotypes. This will be conducted in 7 centers, all different locations, and will be screening with polythene cover and screening for different genotypes using the facilities, sir. And fourth trial is physiological characterization of selected rice genotype multiple levatic stress tolerance. This trial will be coordinated by Cuttack at CRRI. And the fifth trial, screening of submergence tolerance, is conducted in 6 centers and will be coordinated by Cuttack. And the last trial is screening of rice varieties for tolerance to low light stress. These are the 6 trials, sir. Just we'll look at them in detail, sir. Phenotyping of elite rice cultures, these will be studied in 2 factorial design RPD with 3 replications. And these are the indices, some of the drought tolerance indices, which we would like to evaluate and come up with meaningful data for identifying good donors. As far as heat is concerned, sir, we are screening for using light translucent box to identify genotypes which are tolerant to high temperature, especially to create chalky grains. The fourth trial is the physiological characterization of multiple levatic stress tolerance. We have salinity tolerance, osmotic tolerance, and also anaerobic germinations. These three trials will be taken up in multiple levatic stress and will come out with meaningful donors for multiple levatic stress tolerance. The fifth trial is screening of submergence tolerance. This trial is conducted purely by Cuttack, coordinated by them, and it will be conducted at Coimbatore, Titabar, and Pattambi. And we'll also take good data for this. We'll find superior genotypes as compared to FR13A. And there was suggestion that we should include 21 days as a screening for submergence tolerance from 14 days, sir. This also we'll try to incorporate. The last trial, sir, which we would like to emphasize is screening for low light stress tolerance. This we have Swarnaprabha as the variety, and we are now in the pipeline. We have some more varieties which are tolerant to low light stress, and we'll be identifying suitable donors, and we'll be studying the physiological mechanics involved in the low light stress tolerance. So this is all what I want to share with you, sir, as far as physiology is concerned. Thank you.
Thank you. Very good presentation and quick presentation, good trials. Here, only my concern is that you constitute a trial like high temperature and others. It's very good, these are, and actually your result will be useful by the breeders. But here the main concern is you have to the initial population what you are screening should be enough so that the breeders and geneticists they will be able to use in the mapping. So for mapping of the trait for jivas, other thing, These are required actually. So
Yeah, prepare
that-
large populations.
Yeah, the breeders they can merge few years different initial population and can make a representative one. Thank you. Any question from anybody?
Silica trial you have dropped.
Silica dropped.
Silica dropped.
Yeah, dropped. Silica we have dropped, sir. We have conducted for large number of years and we have come out with all the mechanisms involved and we'll bring out a technical bulletin in relation to that, sir.
Okay.
Thus, sir.
Good trials. Oh, you have.
Sir, regarding multiple abiotic stress, we were telling what are the stress and whether it is that along with the biotic stress or only abiotic stress,
Only
what
abiotic.
are the stress you are imposing?
So in multiple abiotic stress, we are imposing salinity stress, water stress and anaerobic germination.
Okay. So prod, anaerobic germination and salinity.
Salinity.
These are the tests.
Yeah.
Okay.
Not flood, drought.
Water
Drought
stress.
also.
Water stress.
Water stress.
Water stress.
Okay. Fine. Thank you.
Okay.
Some Mridul. Mridul
Yeah,
का time
Dr.
का problem.
Mridul, quickly. Yeah, give microphone.
Sir, I have just two suggestions. Even we need certain kind of genotypes which are tolerant here and in biodextrous trials also. There should be some, we also, if there is some common set of genotypes and such kind of genotypes is identified which is tolerant to multiple biotic stress and abiotic stresses, and that will be, if there is some kind of compilation of data from these two sections.
If you have any such material, you can nominate.
Yeah. Okay, sir. Sir, another is this low light condition. Only they are taking the yield data. That is okay. But in the low light condition, a major problem is also the etiolation, that the plants get too tall and only recording and lodging incidences, because if you get any genotype which maintains its constant on the low light, it will be very important. It is especially important for the breeding of the northeastern and the eastern Indian parts. So in addition to this, kindly record the data, that the lodging incidences under low light, and if any genotype is coming out stable as a lodging resistant under low light condition, that will be actually useful for this, and this data is not being
Okay,
generated.
very good suggestion.
Yeah, we will incorporate those suggestions, sir, and we are not only taking yield data, detailed analysis were done using chlorophyll, which is more important, chlorophyll A, B and total chlorophyll. So likewise, we'll try to understand the mechanisms of low light tolerance.
Thank you. Thank you, Dr. Raghu.
Let's, Dr. Raghuveer Rao will be superannuating shortly.
Okay.
Let's all give a big hand for the contribution. And of course, we are also losing another eminent physiologist from our system, who is none other than our co-chair. Let's also give a big hand to him.
Dr. Dave. That we will be doing in the closing function, I hope.
That's right, sir. But this group, we have to acknowledge that.
Okay, thank you very much, Dr. Raghuveer.
Thank you,
And,
thank you,
I
Dr.
invite now entomology PI, Dr. A.P. Padma Kumari.
Sir, one small announcement, sir. Volume 3 is available outside. Physiology, agronomy and soil science. Volume 3 is available. Those who are interested, they can take it.
Okay, ठीक है. आइए मैडम. Routine trial need not to be explained. Any changes you go on telling.
Yes, sir. Dignitary is on the dais and off the dais. Today I'll be presenting the entomology trials technical program for Kharif 25-20, Rabi 25-26. Total of 24 trials are there from ICAR-IIRR. There are 2 trials of NSN from CRRA. In Rabi there are 8 trials. I'll quickly go through. The trial on prophylactic management of plant hoppers is discontinued, and a trial on evaluation of entomopathogens for sucking pests is reconstituted. And we have pest-specific trials, that is plant hopper screening trial at 15 locations, gallmate screening trial at 10 locations, leaf folder screening trial at 22, stem borer screening trial that is at 23 locations, and multiple resistance screening trial with 27 locations. Then national screening nurseries, NSN1 at 19 locations, NSN2 at 17 locations, NSN Hills at 7 locations, NHSN at 12 locations, then Galmich biotype trial at 13 locations, plant hopper special screening trial at 11 locations, Galmich population monitoring trial at 6 locations, then plant hopper population monitoring trial at 6 locations. Then seed treatment for management of early season insect pests at 13 locations. Evaluation of drones for spraying of agrochemicals, that is EDAPM, is at 7 locations. Here, there was a suggestion to include an untreated check, that is a placebo, which we will be implementing, sir. Then bioefficacy for monitoring of insecticides for blind hopper, it is 5 locations have opted. Now there is an increase in number, maybe it will go up to 8, sir. Then influence of establishment methods, this is in association with agronomy and plant pathology, 15 locations. If wherever there is an agronomist, the observations will be taken. Then we have the pest incidence in natural farming with soil science department. It will be taken wherever soil science scientists are there in 14 locations. Then evaluation of pheromone blends for insect pests at 16 locations. Population dynamics of insect pests and natural enemies in rice ecosystem at 33 locations. Then we have an evaluation of entomopathogens against lepidopteran pests, EELP, that is at 14 locations. Evaluation of entomopathogens against sucking pests. This is the new trial at 5 locations. Then integrated pest management in direct seeded rice. This is in association with agronomy. Wherever there is an agronomy, this trial will be taken at 13 locations. Then population dynamics of insect pests through light trap collections at 33 locations. And during Rabi, we have this temporary screening trial at 4 locations, MRST at 2, NSN Boro at 5 locations, NSN ETP at 7, evaluation of pheromone blends, EPBI at 3 locations. We have PINF at 1 location and PDPNE at 1 location. This way we may think whether to continue or drop because of 1 location each. Evaluation of entomopathogens at lepidopteran pests at 2 locations and integrated pest management in direct seeded rice, 2 locations, sir. So thank you. But I invite all the scientists to contribute for the evaluation of against insect pests and strengthen the HPR program. So more nominations for evaluation and identifying the donors is requested with proper germination of the seeds, sir, so they can send us seeds for evaluation, sir.
Thank you.
Thank you, sir.
Madam. Very good presentation, clear. So you have also discussed with your group already.
Yes.
Any other people, those who were other than the group, entomology, pathology, they may raise their question.
And one more suggestion was there in MRST entries should be evaluated for grain moth, sir, that we are implementing, sir, in the technical program.
So no, all are happy with the things. Yeah, one suggestion by the director.
Madam, as a team, both entomology and pathology are doing outstanding work.
Thank you, sir.
For example, at least 2 trials wherein I see there is a good value for making an international publication. One is influence of establishment methods on probably sucking pests you are collecting. And at the end of, say, 3 years or whatever the periods this thing, it should come out as a joint publication from all the centers who are conducting the trials.
IPM,
and,
sir?
Not that IPM. And
IEMP,
yeah,
sir.
and also the trial on natural farming.
Yes, sir.
It should culminate into a good publication.
Fine.
Because there's a lot of controversy. As ours is one of the largest group, and if we communicate that, okay, with regard to reduction in insect pest population, whether natural farming or whatever may be the cultivation practice, whether it is working or not, a clear message has to be conveyed. And what better than AGRM? And whatever support that is required, even it is in a high impact factor journal, if it is on payment basis, we will meet it from continuous.
One request to the scientific fraternity, sir, they should disclose what formulation of natural farming they're using. Let them write down, sir, whatever is Dasagavya, Panchagavya, whatever it is, what formulation, where they have procured, because each matters when we are compiling. Yes, sir. Individually we'll do it, sir.
Thank you very much. Good presentation.
Thank you, sir.
I invite
Sir, Madam, when you are conducting this bioefficacy trial, whether it is a request from the company side or we are in voluntarily we are doing?
We are doing on our own, sir, for our own understanding, because there are many control failures in our management trials. People are asking us. So this was contemplated since many years. Then finally we took a call and we have initiated the trial in Kharif 24. No company is there, sir. We have constituted on our own, sir.
That again there is question. When we are, that product is a registered product. Whether we are picking that product exactly if the company will raise question on that, you are telling that our product is not working, and at that time this IPR issues will come.
So
So
we
that
have,
is to be also taken into account, because
we'll take into account, sir. We're only putting the common name. We are not putting the registered trade name in the report, sir. We are sending it from us
It's
and
very
the product.
concerning. This
This
is
is-
relevant
this is a-
question.
Yes, sir.
This is a great concern.
We'll discuss about it, sir. We'll come up with it. We'll take into
we
account.
should
sir.
not take like this.
Yes, sir. Thank you.
Okay, thank you. So last presentation of this session, EI pathology, Dr. M. Srinivas. Srinivas Prasad. Yeah, please. Only the changes, important
what is the
points
changes are there?
Only show the
Respected
slides
chair and
don't
co-chair,
say
sir.
all.
You have anything to scroll in? Yeah, I can scroll. What will you do? Yeah, word file will be fine. Or I'll scroll. Yeah, these are the respected chair and co-chair, the dignitaries on the dais and off the dais. Actually, in plant pathology group, we have discussed it thoroughly about the trials to be constituted for the coming year. We are conducting the trials under three major categories, sir. The trials on host plant resistance, the first category. The second is on field monitoring of virulence of two major pathogens. And then third one is disease observation nursery. And then fourth one on disease management trials. And this is host plant resistance is a major program for both pathology and entomology. Here we are evaluating the material and IVT and AVT material and also the donors and then hills material across the locations against these six major diseases. These are the leaf blast, neck blast, brown spot and sheath blight, sheath rot and bacterial blight resistance and resistance to the rice tunguru. So we have proposed the trials across the locations, sir. And then under field monitoring of virulence, we have two trials. One is field monitoring virulence on Pyracleria oryza and then Xanthomonas and BLB virulences. Here we are incorporating not only the differentials, we are incorporating the pyramidal lines, single gene, two gene combinations also, so that we get a data in which zone, against the 7 zones, which zone, what are the genes are effective, that we are also observing, sir. Under disease observation nursery, there is a light modification we made it last year that now the disease scenario we are not knowing that in case of now DSR is coming up very well. So we wanted to know the disease scenario in case of transplanted versus direct seeded rice. So here we have taken up these trials across few locations wherever it is manageable. Under disease management trials, we are taking up these trials using the fungicides again location specific trials. Generally, we are taking up here the combined molecules, not the straight molecules, so that they will get a good product across diseases. And then another trial is on biocontrol aspects, sir. So we have formulated one new trial here with a molecule that is identified at ICAR-IIRR, and then the two formulations, most bacillus and then Trichoderma aspergillum, and also the consortium product we are keeping it here, sir, and it was formulated against six. It was to be conducted at 7 locations. And one more trial. This trial is actually it is a combined trial of entomology and pathology and also agronomy, integrated pest management, direct seeded rice here also. So here the trial they will also they raise the trial agronomists and then we record the data from both entomology and pathology. Another trial is a special trial on yield loss assessment. Earlier we have taken up the yield loss assessment trial on BLB, blast and also sheath blight. Now this was asked by our group, then we have taken up last year's of this trial and results were encouraging. A little modifications our group suggested that how to proceed with that 50%, 100% and disease, different ratings of the disease. There we modified a little bit that we will first inoculate the disease and then we spray it twice, first 1 time, 2 times and 3 times. Then we take up the different PDA and then we correlate the disease with the yield loss. Another trial, false mud, sir. Actually, false mud now is a major disease. It has become a major disease. Now we are conducting trial on the identifying good donors for this disease. Our scientists at ICAR-IIRR has identified some lines and then it has been given to our cooperators at few locations. Wherever it is possible, they are inoculating artificially and then to identify a good number of donors for disease. Another trial, actually it was a recommendation from our last year PAMC that drones actually to be used in this. So nowadays drones are being more popularized. That's why we have taken up this trial along with the agronomy and then entomology group, sir. Our group actually, we had another program. It is not the trial. We have a production-oriented survey with the other groups like entomology and then breeders and then agronomists. So this is in nutshell about the programs what are being conducted. And then the proposed, I don't want to read the number of centers, sir. So because of first day of the time. So these are in nutshell about the programs what is to be conducted in the next year, sir. Thank you.
There's
Thank
only
you,
one suggestion. Yesterday, in production-oriented survey, we were missing centers in Orissa state.
Yeah.
The final,
Sir,
this thing,
yeah.
we have included
Yeah.
those
In
centers.
the yesterday there was a suggestion from the CRRA, sir, that they also wanted to include in this program. So we have incorporated Cuttack Center, NRRI, and then also Jaipur we have included. Yeah. No, actually, sir has explained yesterday.
It is taken care.
Yes, taken care,
Told,
sir.
no?
It was already there in the trial now. It will be your data sets will be sent to you, sir.
Dr. Mahapatra already. He is agreeing with what to discuss.
Okay, another issue I had read yesterday, that one of the major concern is the Bakane disease. So once we are going for any evaluation and DIC, whether it is resistant or tolerant or moderately resistant to Bakane disease, in NSN that is not finding the path. So in eastern India as well as in the northern India, so in Basmati and non-Basmati, this is a emerging as well as emerged. We can say it is a emerged problem. But you have, even in spite of that, you have not included in your technical
actually,
program.
the session yesterday only evening you have given, sir.
That's
we will
okay.
include it. One thing, sir,
How much
actually,
time?
IRI we have already included in as it is a very problematic.
Whether you are now ready to include, it's already okay.
It's okay.
We will include and then put Cuttack as a nodal center for evaluating the Bakane for non-Basmati race because IRI is taking care of Basmati.
Any other questions?
Then I will put you that.
Other questions?
Okay.
Sir, go over, sir.
Other question? No other questions. So very clear presentation.
Thank you.
So I hope it is clear to all. No objection further. Thank you. Thank you very much. Now it is overall presentation. Now I say to my co-chairs to say very brief 1 minute.
Thank you very much, Dr. S.K. Pradhan, chairing this session. My colleague, Dr. Sundaram. All presentations are very nicely, they have presented the technical program of 2025-26 and it is well discussed previously in the group meetings and hope they will take care of all those suggestions and concerns raised by the PIs and the chair, co-chair and hope this technical program will be implemented 25-26 properly in different centers and hope a good success in this program. Thank you very much.
Dr. Sundaram, please, 1 minute, quickly.
I'll just take half a minute. Program has been going on really well, but I would like to personally thank our chairman on behalf of you because almost after 10 or 12 years, we now have a separate session to discuss about technical program, not only in individual groups, but as a common group. And sir, your advice was really wonderful. And then it is benefiting us all because earlier technical programs were restricted to the particular group, but all need to know what is happening. And sir, thank you very much for including it, sir.
Thank you. So last also, I will take 1 minute only. So it is a very good, successful discussion. All the trials, particularly breeding, we discussed here very important suggestions from the participants, and they also from coordinating side, they are agreeing to include that. It's a very, very good situation. 1-to-1 we are getting feedback and we are accordingly prepared to test the materials. So very good progress. I hope we have planned well and let us see next year. So that will be our evaluation and all the things, materials, checks, all whatever we suggested are being included and will be executed here.
So thank you all, thank you very much. We took 45 minutes more. That is a thing, and we have to accordingly. Before lunch, I request the other chair and co-chair, so to prolong that till lunch break, so that we will be able to finish. We consumed 45 minutes more. Thank you. Thank you very much.
Thank you so much, sir. So going on quickly, I request Dr. MBB Prasad Babu, sir, convener of this program, to please felicitate our chair and co-chair. We'll do it quickly. So thank you, chair and co-chair, for chairing this session. Dr. S.K. Pradhan, sir, ADG, FFC, ICAR, New Delhi. Dr. R.M. Sundaram, Director, ICAR-IIRR, Hyderabad. Dr. M.J. Beg, Director, Acting, ICAR-CRRI, Cuttack. Thank you, sir. Thank you very much. Thank you, Chair and Co-Chair, once again, and all the presenters for this session. We'll have a quick tea break, and we hope that all of us we assemble 11:30 sharp inside the auditorium, please. Because since we are running short of time, so it's a request that all of us will quickly assemble inside till 11:30.
Hello, 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 check, hello,
Sometime I
hello,
receive your
hello,
lecture
hello,
about publication.
hello, 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 hello. Hello, hello, hello. Please speak out.
This time because we're pressing. इस बार हम लोग का टाइम ही कम कर दिया 1 घंटा
Sir, good morning. आपको सुनने के लिए आया हूँ।
आदत खराब हो गई इसलिए कि कोई असर नहीं पड़ता ना नहीं खूबी नहीं असर पड़ता नहीं फिर वो लिखेंगे ना डीडी जी को लिख देंगे पहले वाला को तो प्लान फीडिंग समझ नहीं आती थी तो प्लान फीडिंग था ही नहीं कभी खेत में नहीं गया ये तो आदमी खेत में उसको फिर भी ये क्या जानता है मुझे तो क्या जाना చెప్పలే చెప్తాను చెప్పు
So now we begin with technical session 3, which is a special session for platforms and collaboration. For this session, we call upon the chairperson, Dr. B. Mishra, former VC, SKUAST Jammu, and the session will be jointly chaired by Dr. Sudhanshu Singh, Director, IRI India. The co-chair for this session is Dr. B.N. Singh, former Director, ICAR-IIRR Cuttack. So I request that the chairs and the co-chair to please occupy the seats on the dais. Conveners for this session are Dr. R.M. Sundaram, Director, ICAR-IIRR Hyderabad, and Dr. M.J. Beg, Director Acting ICAR-IIRR CRRI Cuttack. Rapporteurs for this session are Doctors M.S. Ananta and P. Sengutwel, ICAR-IIRR Hyderabad. So I request the rapporteurs also to please occupy the seats on the dais and record the proceedings. Over to you, Chairman, sir.
Good morning, everybody. On the dais, I see very important people holding expertise in their specific field. So I welcome you all. Dr. Sudhanshu Singh is there, Director, IRI South Office. Versatile Director of the Institute, Dr. Sundaram. Dr. B.N. Singh, who loves rice, I think more than himself. So, welcome you, Dr. Zach, and reporters. I see many distinguished people in audience, Dr. Singh, many more, scientists. This is a very precious year, you know that, when we are celebrating our 60th AGRM program, along with Diamond Jubilee celebration. And a lot we have heard in the previous sessions. In inaugural session, very nice presentation. And I think Dr. Sundaram has made very crystal clear how the things advanced, what is the recent going on, what is the achievement, and what's the next in future. He has outlined critically. I congratulate you, Dr. Sundaram. And I think we will not take time. If you all agree, we can give our remarks at the end. So, first speaker is our Dr. Satyaranjan Das, honorary professor. The time allocated is 20 minutes. So, 20 minutes. So, I will request Dr. Das, kindly limit within your time. Dr. Das. Okay.
My dear revered teachers and my dear young friends, and this is a unique opportunity to share my long years of experience in rice research. And I choose the conventional breeding, and we will start now. I should not waste my time. because time limit is only 20 minutes. So by the adoption of the semi-dwarf rice varieties, coupled with improved management, there was spectacular improvement not only in yield but also we have achieved self-sufficiency and surplus. And by the incorporation of a series of resistant genes conferring resistance or tolerance to biotic and abiotic stresses, the breeding populations, the yield level has been stabilized. Now I would like to tell you this one, that if the experimental yield is 100%, the enterprising farmers, they used to lose 70%. And the actual realizable yield at farm level is 30%. And there are many constraints about the variety, constraints, time of planting, modern practices, drainage, weeds, pests and diseases. But for the yield gap 1, the main constraint is our risk-free input management. Then the rice production in India recorded a steady upward trend during the last 5 to 6 decades with almost four-fold increase in production. That is from 34.5 million tons to 137.8 million tons in 23-24. And due to enhanced rice production, the country became the largest exporters of rice and earning about more than 86,000 crores during 23-24.
कॉलम में लेंगे तो आराम से क्लिक करके कर सकते हैं सर इस 2 मिनट आपको थोड़ा आगे पीछे जाके
उसको करना होगा
सॉरी सर ठीक है आप मूव कर सकते हैं
So at the current level, per capita availability of rice production is required to go up by 40% and 70% from 137 to 180 million tons and about 232 million tons by 2030 and 2050. So achievement of such a huge target, and we shall have to get this one next few decades as the increase in productivity has to come from the declining and degrading resource base, and also there is always a demand for environmentally sound rice production strategies. Then there is loss of genetic diversity in rice. There were 4 waves of loss of that. When we will go back our pure line history, that 82 research stations which were spread over 14 states developed nearly 445 rice varieties through pure line selection by improving native varieties and with highly heterogeneous native farmers variety. And here mention may be made of GB24. That's a pure line selections from Konamuni. Why I have given this one? That has contributed lavishly in the national as well as the international rice breeding programs to transfer this better grain type as well as the resistance from GB24. And the next depletion of variability due to the wide adaptation of the semi-dwarf rice varieties, leading to a sharp reduction in the genetic diversity. And the third one was the hybrid approach, by the use of the same sterility inducing wild abortive cytoplasm. And the fourth one, there was increased emphasis of molecular marker assisted backcrossing, and there were also few mega varieties were there. And there is also that brings down genetic diversity as the fourth wave. Now, the important rice improvement programs, including the International Rice Research Institute, we have not made any significant achievement in terms of the yield potentiality after IR8. And if you will fail to stabilize the yield growth, at least where we stand today, then it may not be possible to sustain our self-sufficiency and surplus in rice. But the plant breeders argue that there is no yield plateau. A perfect variety has not been evolved. Still there is an unexploited genetic variability for improving the direct and indirect component of yield through the utilization of the untapped germplasm. There is still scope to develop and use more efficient breeding and selection strategies. So we start with the mating system. Then in the biparental crosses were used widely to serve the purpose of combining traits from the two parents, but the recombinations from two parents is too restrictive, and the use of multiple crosses was suggested by Harlan Mattini in 1940, involving 16 to 32 parents, and they are crossed in successive generations until the final hybrid involved all the parents. Theoretically, the multiple crosses provide an opportunity for recombining of and providing opportunity for recombination of genes from several sources. And you can see the flow chart. There are here 8 parents used. There were single crosses, double crosses, quadruple crosses, and final hybrid contains all the parents. It seems very easy, but it is not easy to make crosses, single, double, quadruple crosses. It is, but the major limitations will be, it is practically not possible to get enough F1 seeds in the latter generations of mating to retain all the parental genes. So, assuming multiple crosses involving 16 parents, each having only one favorable gene, the number of hybrid seeds required at 50% probability would be 8, 64, 131,000 and 64,000 to the power 4 in the first, second, third and the final generation respectively. And another drawback, involving 16 to 32 parents, there is every likelihood that some parents were included into the breeding populations and with to disrupt the good genetic background of the varieties that took several years, even decades to assemble. So therefore it was suggested modified backcrossing by Mackay in 1954. Then the 3-way crosses were tried in many selfing species, and the third parent is another adaptable germplasm, which serves the purpose of increasing the proportion of the adaptable germplasm and simultaneously widen the scope of combining desirable genes from another good parent. And it has been observed that the three-way crosses have greater genetic variance than the two-way crosses and produce more superior recombinants than the two-way crosses. And such crosses also provide an element of gamete selection as it was suggested by Stadler. The F1 plant reflects the combination of the superior gametes from the segregating single cross and with the genotypes of the non-segregating variety as the third parent. Then, as argued by Stadler in 44, the frequency, frequency of the superior frequency of the gamete is much higher than the superior zygote. Therefore, three-way crosses provide a more efficient mating system for obtaining lines with better performance. Also, double crosses were tried to extend the range of parents to achieve recombination from desirable alleles from diverse parents. So, this different mating system, as suggested, was limited to a great deal of requirement of the large number of F1 seeds. And the F1 generation serve as similar to F2 of single crosses and said the limited possible recombination of genes available in the breeding populations. Nevertheless, by the use of these multiple crosses to create an array of genetic variability that never existed in any breeding population. And many rice breeders have not used multiple crossing programs and because they got the adequate success by successfully manipulating and carefully manipulating the biparental progenies. However, we find there is the requirement of large number of F1 seeds, relatively small sample size in F2, which is usually used by the breeders nowadays. A majority of the plant breeders, rice breeders have restricted their selection to known material and have made intense effort for local adaptation. As a result of certain gene blocks, they get rapidly fixed along with correlated response, which in many cases may go in adverse directions. Therefore, it is suggested that we must have to choose parent with wide genetic base, with acceptable level of productivity and high general combining before they are used in a multiple crossing programs. Then about the gene pool concept and use of weedy relatives. The gene pool concept for plant breeding was conceived by Harlan and David in 71, which divides the whole array of genetic variability within the reach of the plant breeders into the primary gene pool, secondary gene pool and tertiary gene pool. Often the primary gene pools include the progenitors of the cultivated rice that can be easily crossed with the cultivated types, produce fertile hybrids and exhibit normal segregation. And one example here, that using the progenitors from the primary gene pool, that is very exciting result. Frey and his group over a period of 20 years, that in oat interspecies mating, Avena sativa and Avena sterilis, with 3 back crosses, they got 3 to 29% yielding improvement. And the introduction of germplasm of Podium spontaneum into the cultivated barley also increased to the extent of 29 to 46%. And there was a very good level of agronomic traits realized in back cross 3 generations, 2 back cross 5 generations. These interspecies mating take the advantage of nuclear genes, and to what extent the wild genes can be tolerated, and this is about 6.25% or less of the wild genes can be tolerated both in wheat and barley, and can tolerate about the same percentage of wild germplasm for expression of improved productivity. So both the cultivated there, when we'll come to the rice background, both the cultivated types Oryza sativa and Oryza glaberrima, and they have the progenitors related wild species like Oryza rufipogon, Oryza nivara, Oryza brovilligulata, Oryza longistaminata have the same AAA genome and they share a common gene pool. These weedy species can be easily crossed. F1 hybrids have normal chromosome pairing but show the varying level of sterility. More often, these wild species, they are not very frequently used in the crossbreeding program by the plant breeders. And usually used for transferring resistant genes. We all know about the story of Oryza nivara and IR36 and IR64. And it must be systematically exploited for the improvement of wheat. Therefore, it is suggested to make use of
5 minutes only.
Wild species from the primary gene pool and examine the feasibility of wild germplasm for improving the productivity. Then the disruptive mating was tried, and here you can see the where there will be plus and frequency distributions, they were intermittent, and as a result of which in a predominantly self-pollinated plant, only the in the state of heterozygosity in F1 there will be crossing over and release of variability which is locked up by the repulsion phase of linkage. But this process will enhance the genetic variability. So male sterility facilitated composite. And actually this is a method of breeding which initiated by Sunesan, and the Harland variety was the product which was the 28th generation seed lot of the composite cross bulk. And natural selections had caused the bulk populations to become higher yielding in later generations. Therefore, now you make crosses, involve large number of parents in the crossing program. You should not follow the pedigree system of breeding, but you opt for bulk population of breeding. The bulk breeding is to handle large genotypic variations available, and nevertheless, by artificial selection, the bulk populations can also be improved. Natural selection is crucial for the evolutionary process, and there is a strong selection feature for characters of high adaptation. Higher recombination by breaking of parental linkage groups may help to form a better base population. Now the male sterility, monogenic male sterility facilitated composites has been exploited for many objectives has been solved. And the recurrent selections also tried in corn for 100 years, more than 100 years, in wheat, in China more than 30 years, in barley, Canada, and in rice, the mainstream of breeding in upland rice in Brazil, and the recurrent selection is the process of intermating of the selected plant to create heterozygosity and release of variability which is locked up by linkage. So here the selection of a group of genotypes that possess favorable genes, mating among them, and release of variability which is locked up by the repulsion phase of linkage. And this I am not explaining. Many male sterility facilitated recurrent selections has been done, and male sterility facilitated recurrent selection projects For a tropical upland japonica, lowland tropical japonica, indica, and etc. they were done by the international effort as well as IRRI, WARDA, SEAT and CR. And there, these composite populations were distributed to the plant breeders, where they will have their local selections and developed varieties where these populations, they contain many parents and diverse parents here. And about the male sterility, that the genetic male sterility, there is a disadvantage. It has a selection disadvantage in the population, and the frequency decreases to a very low value within few generations, and this effectiveness of this system becomes soon lost. And there is also some evidence that too much crossing over in case of a predominantly self-pollinated cropland, which is detrimental to the fitness. Then DSF systems, everybody knows that how it can be used now. At every stage the varieties can be there, and finally a dynamic gene pool concept will be there. Then the history of stay green indica germplasm. This was in the variety was Federos 50. So as there is no time, I will tell you one thing here. So the picture that this Federos 50 was with senescence inhibition gene from Rhabdopsis were transformed with extraordinary increase in photosynthetic rates and chlorophyll content. Gains in tiller number, seed set, plant yield and biomass. And the flag leaves of the stay green Federos 50 at grain maturity showed large advantage in chlorophyll content, photosynthetic rate and nitrogen content. You can see this is a senescing variety and this is a stay green variety. So therefore, why not? Through conventional breeding program, we must use such varieties in the crossing program and study the physiological aspects of the stay green trait and make improvement in yield. Then the proposed research, I am leaving this one, no time. Then the development of the heterogeneous populations. This was, sir, please. So what happens, there are some instances that a variety T141 and its pure line counterpart is, the local land race is Saruchinamali, and its pure line counterpart is T141. And similarly, the another variety that is Muja Bukulu in Andhra Pradesh, Kartika Isamba in Tamil Nadu, and their pure line counterpart were BCP1, SB7 and T141. The farmers, they prefer the land races rather than their pure line counterpart. That means we shall have to, not in conflict of the pedigree system of breeding, and it is possible to create a heterogeneous population so that for greater stability of performance. And this was a part of my PhD program at ICAR-IIRR, and here you can find here मैं दिखा दूंगा, इसको रख दीजिए. So what will happen? The different lines were developed through pedigree system of breeding, and now on the basis of phenotypic similarity, at least with respect to grain type and height, we can blend it in different proportions and make a composite population. And there, our idea is that these composite populations are expected to be not only higher level of produce, but also they will be stable over years and over locations. Now, the story of Sub-1. Here you all know that the sub-1, which is a major QTL contributing about more than 70% of the phenotypic variability, has been successfully introgressed into the different mega varieties. But under normal conditions, we find, as flooding is not a recurrent problem, we find there only few are better than their recurrent parent, and therefore what we did, we must, I have some photographs, and here you can see this is FR13A from where sub-1 was identified, and the SN sub-1, IR64 sub-1 and Taiwan sub-1. So in terms of survival and regeneration, you find there is difference. So the question arises, although the same QTL has been introgressed, and why such variation is there? And that is primarily the background history of the recurrent parent. The IR64 sub-1 was best in terms of survival and regeneration, because we all know that Dr. Khush took so much effort by convergent breeding, incorporating large number of diverse genotypes for the development of the IR64. Now, we find sub-1s, they were under stagnant flooding, are not promising. Therefore, we have taken to converge molecular breeding with that of the conventional breeding to integrate, along with submergence tolerance with the stagnant flood. Then there are some traits, all breeders they know, I am not going to tell this one. Then the nature of character association, also not telling. But remember one thing, yield is a function of dry matter and harvest index. And for improvement of the yield, either we shall have to increase the dry matter or the harvest index or both. And here we must remember Professor LP1's statements. That
Doctor, please conclude. Already 10 minutes over. Half an hour you take.
2-3 minutes give me. So this is the LP1s where I am interested about the leaf angles there. Flag leaf, second leaf and third leaf, where there will be the radiation utilization efficiency will be very high, and as a result of which we will yield that the productivity can increase. And this one I cannot skip. What happened here? Two traits, 100 grain weight and fertile grain number was plotted. When the fertile grain number was there, the frequency of the yielding genotypes that increased. But you will find where there is a balance association, a large number of genotypes were accommodated. And I used to tell this one, that highly intelligent people, they do not survive in the nature. And dull people also, they do not survive. And those people, but there is balance association of traits, they survive and they show a greater stability of performance. Now, the convergent breeding strategy, that gradually, step by step, we will incorporate traits. And remember, भगवान के घर दे रहे कभी अंदर नहीं है. And gradually, finally, we get a genotype with high yield and better genotype. Then the rapid generation advances and the speed breeding, where the empirical formula, which is used by the plant breeders, that is selection intensity, heritability coefficients, and phenotypic standard deviation. But for speed breeding, divided by L, which is the length of the breeding cycle or generation interval. And when we reduce this generation, our genetic gain will be high. Then comparison of different pedigree system where we can use, bulk where we can use, and considering the cost of the line fixation associated with RGA, SSD was found to be effective than the other methods, which is less costly and more rapid. Then the anther culture derived dihaploids were also used for making improvement. Whatever it may be, the superiority of any breeding method is largely dependent on the choice of parents, genetic variance for different traits, sample size, selection accuracy during the segregating generation, and environment interaction, genotype environment interaction, genetic drift, and changes in the gene frequency due to inbreeding and heritability of traits. And many methodology has been tried. Now we will restructure and then the use of new technologies, then the molecular breeding will also continue, and there is synergy between breeders must be there about the molecular genetics, and there is knowledge gap and application gap. It is interesting that many times the breeders, they do not the intricacy of biotechnology principles, and sometimes also we know the biotechnology, they cannot understand this one. So there must be, for greater use of efficiency, there must be synergy between this one. And in future, there should be planned and well executed QTL mapping, QTL and marker validation activities, efficient genotyping systems, cheaper genotyping systems, and breeder field analytical tool. Integration between breeders and molecular genetics will have a new germplasm. Where my teachers, revered teachers, he is Professor Mahanti, H.K. Mahanti, who was the main architect of rice breeding in OUT. He is no more, and he is the legendary Dr. E.S. Siddiq. At any moment you reach to him, a new emerging problem and its solutions will come. And this is an opportunity to work with McGill. And this is a very pleasant moment to get a Senandira Rice Research Award. And my dear young friends, remember, in difficult moments, behave like a dog. Keep calm and unruffled on the surface, but keep paddling away like crazy underneath. This is harvesting joy, which is a gentle reminder that life is a cycle. What is sown, it must be reaped. And my young plant breeders, while there are indeed many difficult challenges ahead in the 21st century, that we must remember that even 1% increase will be a tremendous impact. Even in
That's enough.
Okay, sir. Okay, sir. Okay, sir.
Thank you very much for nice presentation. We know that you are very rich in traditional wisdom and very systematically you have presented.
Sir, you see,
So
nothing is
I
more.
will invite the question after all the presentations are over.
Okay, sir.
Thank you very much. So our next speaker is Dr. S.K. Pradhan or Vilved ADG FFC, and his topic is genomics assisted breeding for rice improvement. Very close to his heart, this topic.
Thank you, Honorable Chairman, Co-Chairman, rapporteurs, my teachers here and friends. You heard now one very emotional talk and classical breeding. This was my suggestion actually when I was discussing with Dr. Sundaram. This is a it's a golden jubilee year. We should include few topics that will be better for our collaborators. What is the status of the conventional breeding as well as how far we are in the molecular breeding part and also in genome editing along with how far we are in registration, what is the process of registration. If all these things we will be able to know, then it will be highly useful to our collaborators. So while coming to my topic, you already heard a very good lecture about classical breeding. So my topic is really about what is the advancement here, marker assisted, genomic assisted breeding. Now we are pressing a lot hard last 2 to 3 years. I am always telling you start genomic assisted breeding. Why I will be telling and what are the status, where we are, whether we will be able to practice genomic assisted breeding or not. That let us discuss. I will take less time compared to Professor Das. I will try to finish in time. Let us try. This is a very bigger topic. So my purpose is here. Why genomic assisted breeding we want to do? And what is genomic selection? Breeding scheme for this. Optimization. How we can optimize our pipelines and models what we are using. And finally, I will be concluding. We have achieved our target of more than 10 years ahead. We achieved our rice production about 138-40 million tons. We are increasing production. We are increasing productivity. Slightly, the area is increased. So then, what is the need? Why we are concerned? That I want to say. Just you were listening the lecture of Professor Das, where he was confused. So many, when we will be meeting with so many parents, it is difficult to handle the traits, parents in together and selection of the progeny. This is one approach where simultaneously we can put so many traits together. That approach is the genomic assisted breeding. Here I will be telling you how simultaneously so many traits together you can put in a superior progeny. That is the main purpose. And here I have shown here, the challenges, why we want to go for this genomic assisted breeding. Our adverse climate change, so many effects like drought, submergence, so many stress tolerance we have to think for if you are targeting the rain-fed ecology. So the number of genes and traits here, maybe stress tolerance for drought, submergence. In total, many are there. Similarly, this main victim is our rain-fed ecology. About 40%, more than 40% rain-fed ecology. That ecology we are targeting here, multiple traits together. If a few we are confused, I will be telling what is the problem. And then increasing biotic, abiotic, it is known to everybody why so many traits we are thinking. Then also reaching the power production target like low emission, other things, we are also concerned. And food security we achieved but did not achieve the nutritional security. So many here also for biofortification, so many candidate genes you have to put for protein, zinc, iron. Each they have multiple QTLs. Major gene also. So also suitable technology requiring less investment, young people they are not getting and potential land are being converted to business for business purpose, or commercial purpose. So therefore, what is our need now? We need a variety or ecology based variety, good varieties with multiple biotic, abiotic traits with superior nutrition and low input responsive fitting with pro nature and environment friendly. So these so many traits putting together, it is very difficult to go in a classical breeding as Professor Das was telling, if in a convergent cross, if you are facing so many difficulty, it is difficult to actually go ahead. So this is the right approach where we can simultaneously put in a breeding program all the trait genes together to achieve it. So this is the basic of that. Then what is it? What is genomic assisted breeding? You know, you heard first point, I don't want to say, relying on the phenotypic selection. So due to this phenotypic selection, we achieve the green revolution. But still then, what is the problem? Then we thought about marker assisted. It's good. Many few genes QTL we transferred and we developed the marker assisted products. But the problem is that, as Dr. Das told, if when the traits, number of traits are, we are thinking many more, say about 10 genes if you will be putting together, then in case of marker assisted, you will be phenotyping lakh progeny, millions. Is it possible million you have to phenotype as each segregating generation, back cross generation? So that is a very difficult task. So few genes, maybe 3, 4, 5, 6, you can put together, and marker assisted breeding is very good. What's about small genes, QTLs? Those you effect you cannot know. There are many small QTLs. In those cases, it is the marker assisted. It's very difficult to do. The error part will be here. It's very much so difficult to do. So here in this genomic assisted, our target is not few genes. Whole individual as a concept we are keeping here. So what is the genomic effect of the individual rather than telling a single QTL or few QTLs. Therefore, here in the genomic selection, we are deploying markers to estimate the effect of the individual, not for a particular gene, whole individual. And that effect is estimated using statistical models. So this is the actually basic thing in case of genomic assisted breeding, where you can think of marker and phenotype based selection. Therefore, this is a very advanced one compared to mass, pure, phenotype based. Both are of course here involved. It is the integration of both these. So, you know how why it should be popular, you know very well. It's low cost of genotyping now, and also suitable models with us. Statistical design models, we can analyze the gene effect, and therefore it is one most criteria you have to achieve. That is the linkage disequilibrium. In your marker system, it is assumed that the markers will be in a linkage disequilibrium. So therefore, you have to estimate here phenotype as well as marker effect, and you have to correlate that. What we are telling, that is genomic estimated breeding value. Based on this genomic estimated breeding value, we have to plan our design all the trait together in a crossing plan, and then we will be moving ahead. So, you know why there is a correlation of genomics and phenotype, and linkage disequilibrium is involved. That is the main part is this genome-wide association mapping actually, or linkage disequilibrium. So in brief, I will be moving this is the main principle behind the genomic assisted breeding. So therefore in brief, maybe 3 to 4 minutes, I will be just refreshing you all. So you know always mapping is a very old concept. Where is the location of the gene on the chromosome? That is actually your mapping. And mostly we used to do is usually either biparental, that is commonly earlier we used to do, but now we are using LD mapping, association mapping, or if whole genome is involved, that we are telling genome-wide association mapping. So why it is important, you know, in case of that I am just showing. You see in case of your biparental, you are contrasting, you are hybridizing and mating population, while in case of other one is you are taking the historic recombination. You are a close family recombination you are doing here in case of biparental. What is happening here, you are taking more number of time is very high and resolution is very low. But in case of association mapping, your nature's material, historic recombination variability that we are using here as the mapping population. So therefore here we use to detect QTLs just to correlate genotype with phenotype. This is the main thing in case of association mapping. And here you can do validate whatever biparentally, any through biparental mapping genes you are reporting. First you can validate here, and also as well we can report the novel QTLs. So basic principle I told you, there should be your markers, population should be linkage disequilibrium. That is the best theory here. So you can include here land races, breeding materials, and any all elite materials in the mapping. So it's the basic thing, linkage equilibrium you know. So linkage disequilibrium is opposite to that, and you can find and how to estimate the disequilibrium. Already established formula. So these are the steps already I told you just in a diagrammatically, or I am showing you. Say you have to make a panel. That panel should be a representative one. Actually, from all the groups, phenotypic groups, all the traits it should represent. Neither it will give a false association. And then phenotype and genotype and assessment of LD you have to do. Then you have to go for marker trait association. Then after identification, you can either find that or clone the gene. So here two actually analysis method are used. Either you can use GLM or MLM. But we used to detect the significant association using both the models. So there are software and you can use that. So here I am giving the example what is the diversity, how you will be selecting here. A continuous variation, say for plant height, you have to select such traits which should make a panel. So here you see the box plot where the diversity is very clear. It is taken all the groups. So also here you can see the probability here for semi-deep it is clear peak is taken. So these are some of the publication I want to show you where it is how used for this GWAS to map some of the complex trait. So next, let us come to our again genomic assisted breeding. So here all the breeders, this is the breeder's equation. If it is fitting, then we are getting gain. If it is not, we are not getting either gain, yield improvement, then breeder's equation is not right. So here you see the numerator, all the things are we can change, but here if years or cycle is very high, then gain is very poor. Our gain will be very low. So you see here how then we will be designing one breeding program. Here actually what my suggestion is that select varieties, elite progenies from different breeding program which are very superior and make a base population for the target traits. Maybe what are the target genes, you make a base population, training population. Then estimate the GBV, I told you importance of GBV, and then suitable crossing plan. How you can, as Professor Das was showing, you make a suitable plan so that all the genes putting together you can do that, and reduce the breeding cycle. Here either you have to go through a speed breeding or double applied method to reduce the cycle. Then heritability is very important. Monitor this using multi-locations and multi-year. Then fixed then evaluate in the first cycle. If it is okay, is passing, nominate in the All India trials, if suitable, will be released or enter it to the second cycle. Again, do that. If not okay, then go to the third cycle. So repeat this one. So this is the basic step for this. And this I told you already is in a diagrammatic form. Either base population, training population and breeding population all together you put in the first stage and see if it is suitable as per your multiple traits. If you are achieving all together, then release or else you send to the second stage. And if in the second stage, if it is okay, then neither you have to push to the third cycle. So this is in detail. What is the scheme I want to show you breeding cycle? So you have to take either 200 to 300 elite parents and then cross based on the GVV. You hybridize that. How to GVV, you know, you have already estimated and cross according to all that progeny and make a speed breeding, yeah, DH. So that within a year we will be getting 4 generations. And maybe within 2 years you will be OIT, AYT and release. If not, all the trade what I showed you in the slide, if not combined together, then you may go to the second cycle and go on that BBB again, make the base population and again hybridize and come to the release stage. If not possible, third one. So this is the thing through breeding cycle I used to say you. If you want to select also segregating generation in between in your speed breeding stage, maybe BC1 F1, BC2 F1, yeah, something in between that, you can haplotype base, you can select to reduce the population. Why to advance so many? So you can have a shorter, again reduce the population and more effective precision you can do. You have this haplotype based. So please go through these publications I have compiled, and here how to optimize the pipelines. Here how to go with the this breeding genomic assisted breeding. Just within a few minutes I want to finish. So here if you are going with more number of season, yeah, more number of testing stages, you see your gain is reducing. If you are having more season per year, your gain is increasing. So similarly, if you will be seeing population size, as Professor Das was showing, if you are taking 12, 13 parents, you have to take huge, large population in case of segregating generation. But in case of genomic assisted breeding, you need not be worried. You see here the study, it is seen that ideal population size, you see in the graph, 100 you will be getting this much of gain. If 200 population you are getting optimal. If 300 it is okay, and it is going on 1000 population size, its incremental gain is very low. So your ideal message is it should be 200 to 300, yeah, at least 100 population size. So similarly, how many replication we should go here? You see, if you are taking one replication, your gain is very low. If two is okay, if three is
20 minutes.
yeah, 1 minute more. So, seeing you, I told you for selection you have to go with a marker chip, and that should be like a mid-density marker chip, maybe 5 to 10K. And ideally, if you have a starting program, go with 1K Rika chip. If you are mapping also, you can go with 1K Rika chip. So here, if you have enough resources, you can go with a higher density marker. So models, this is the last part, sir. Model, I want to say, it is the main thing. Unless you estimate statistically your markers effect, you will not be able to estimate your breeding value. So usually all are tempted to use G-blob. But I am telling you, G-blob, what is there, is the average effect of the marker, it is taken into account. So average effect, no issue, but when there is a major QTL, there is a major gene, in that case, it will make an average. So that means you will be able to detect the gene, but effect will be biased. So if you will be going to the next one, regression based, Again, regression based is okay. It will not give a means average value, but it will give variation value. But here the effect will shrink to zero. So that is a problem again to the breeders. So next is your lasso. Lasso means Lysian is least significant. It is the model where your deviation will not be much. But in case of Bayesian approach, actually, you have both this average effect. There is a assignment of value to each of the marker accordingly, and you can have a balanced one. So there are different Bayesian. Even if you have your non-parametric models like random forest, SVM, machine learning, those things, when you will be doing more, when you want to study interaction of the markers, then you will be seeing this. I hope I finished my talk here. Whatever I told, it's just in a summary. You have to multiple targeting, multiple trade together is possible through this, and gain is reduce the length of the breeding cycle. Similarly, your stage of breeding is important, and population size I told, replication I told, marker I told. If your resources is you are rich, you have enough funding, then go with high density, or else you go with like 1K. So thank you very much for patient sharing. Okay, of course, a very long topic, very difficult
Thank you.
to
It's a
precise.
timely.
Yeah, thank
you
you
have
very much.
presented. It's a wonderful presentation. Very good knowledge of modern science. But we'll invite questions and
Yeah, sir.
clarification.
Thank you.
after.
Thank you.
Thank you, Dr. Prasad. So now I invite Dr. C. Vishwanathan. Respected Chair, Co-Chair, Conveners, Rapporteurs, dignitaries present of the dais, delegates. In next few minutes, I will try to cover about genome editing, although the time is short, so I may not be able to justify with the topic completely, but I will try to see like how best I can cover. So you all know that genome engineering is not something new that we have been practicing, and the variation that we see has been possible because of genome engineering. Although the methodologies have been evolving, and if I classify the whole plant breeding into two parts, the first part, which was mostly it was like random genome engineering, wherein we did not have the control over the flow of information or the flow of DNA. While in the second phase, now we are talking, it's more precise, more specific, wherein you can control, the flow of information or the DNA. Coming to the genome editing, it's now I'll not go into the details of its basics. It's basically a precise modification of any gene. It could be within a gene, it could be an exon or promoter or intron. But eventually, what we do is to modify some specific nucleotides or bases, and it has been broadly classified into three kinds of genome editing, that is SDN1, SDN2, and SDN3, that is site-directed nuclease, depending on whether we use any template or not. In case of SDN1 and 2, we do not use any template, while in SDN3, we use template. That is why still it is considered as
so I am sorry, for SDN2, we do use template, but the purpose is only to correct the gene correction, while in case of SDN3, we go for insertion of either part of gene or full gene, and that is why it is still considered as transgenic, or it undergoes through those strict biosafety regulations. In next few minutes, I will try to cover the story of Kamla that we have been doing at Indian Institute of Rice Research. That includes planning, dry lab research, tissue culture standardization. Believe me, 60% or 80%, I would say rather, the success relies on the first component, that is the planning and the dry lab research. Since we were working on Samba Masuri is highly tissue culture recalcitrant genotype. It took a lot of time for us to decode its protocol, and then development and evaluation of these genomated lines, identification of the Kamla mutant that now we have passed through the multi-location testing and now identified as variety. So these are the most critical aspects, particularly if you are utilizing genome editing for the varietal development, wherein the selection of gene, selection of cultivar, and the trait. These three things are most critical, and one has to really, as I said, the planning is most important. Put a lot of thoughts behind before we proceed for it. And in our case, we selected a gene, cytokinin oxidase, which was discovered in the year 2005 by Asikari et al. They were working on two genotypes, Kosi-Hikari and Habataki. The purpose was to map a gene which regulates a number of grains in a panicle. As expected, or so the most I would say advisable pathway is, if we have to map a gene, what we will do, we will develop the biparental population. That's what was done. In case they developed the biparental population of Kosi-Hikari and Habataki, mapped the gene, and they identified the gene that actually degrades the cytokinin, and the gene was cytokinin oxidase 2. Now, what Habataki has, it has got a gene which is mutated that includes 16 base pair deletion in 5' UTR and 5 base pair deletion, 6 base pair deletion in first exon. Now, because of these two mutations, it has got compromised activity of cytokinin, and since it regulates grain number in a panicle, the gene, its form of the gene, that is cytokinin oxidase, it was named as GN1A, or the grain number 1A. Interestingly, if you see in this paper, the Kosi-Hikari and Habataki, the number of grains per panicle in Kosi-Hikari it is 164, while in Habataki it is 306. But they have also mentioned another genotype. When I read this paper again and again, and I was like kind of bit curious that why they are not talking much about 5150, which has got 405 grains in this panicle, and this has got a different mutation. It is not GN1A. In 5150, this particular genotype has got a mutation in exon 3. It does not have any mutation in exon 1 or exon 2. In exon 3, it has got a mutation of 11 base pair deletion. And this particular mutation is exactly at the position where the cytokinin binds to the protein. And due to this mutation, the number of grains in a per panicle have gone from 305 in Habataki to 405 in 5150. What they wrote at the end of this paper, just one line, that a null allele of CKX2, that is 5150, had been selected for increasing crop yield in conventional breeding program in China. And then another paper came in 2015. I was analyzing all these alleles and found that there are 14 alleles, and some of these alleles are localized in in other parts, but they are not there in the Indian germ plot, particularly the mutations in exon 3. So what we did, we did thorough analysis of the gene and made the modeling of the proteins, and then see that if we mutate the cytokinin binding pocket, what kind of mutations we can bring. So we mutated the exon 3 and exon 4 after modeling of this particular protein and developed the best superior allele, which not only enhanced the number of grains, unlike 400 grains in 5150, in this case we have recorded up to 700 grains in a panicle, on an average 450 grains per panicle. Along with this, we got a strong culm and also earliness trait. And this particular allele, we filed the patent to protect because this version is not present anywhere. In order to create this allele, we have created 84 different versions of this gene or 84 different alleles by creating different mutations, evaluated more than 1,000 lines in biosafety screenhouse, and then did the performance analysis of all these lines, and then identified this particular mutant that we now call Kamla. You can see here the plant has got very high vigor and it matures early, very good culm strength, very much robust panicle size. Here is on my this side I am holding Sama Masuri, which was the original version, but after doing the cytokinin oxidase mutation, now you can see the difference in plant vigor. It has also led to increased flag leaf length, flag leaf width, and also the photosynthesis rate that is helping the plant to fill those many grains which are there in the plant. Otherwise, in just enhancing the number of grains would have not helped. This is an analysis wherein we have compared the panicles of Sama Masuri and Kamla. The increase in number of grains per panicle is totally because of better panicle architecture, more panicle branching. You can see here the branch length is also increased and the branching is increased. The grain quality characteristics, which are actually USP of Sama Masuri, they remain intact. We have not compromised with the number of feeders. They are very similar to what. So there is till now we have not actually noticed any significant trade-off due to enhancing the number of grains in panicle. Rather, what we have found, these lines are having better nutrient use efficiency, water use efficiency, high plant biomass, that is also making this line to grow better under DSR conditions. This is what exactly we were trying and what has happened. We did not want to completely knock out the protein because it will enhance the cytokinin level too high, which may not be or which might disturb the plant phytohormone homeostasis. So what we were trying to do is to just increase the cytokinin binding pocket wherein it does not bind the cytokinin very tightly. And now in this case, the cavity where now the cytokinin binds is now big enough that it does not bind the cytokinin very tightly. So there is cytokinin degradation, but the extent of cytokinin degradation has reduced sharply. We have in fact analyzed the level of cytokinin in this line and also the other phytohormones. Because of the cytokinin increase, there is a kind of phytohormones crosstalk, the change in auxin and also change in particularly brachiosteroid. Excuse me. Yeah, so this particular line after identifying, this was the science behind I was talking. We had nominated this line in Kharif 2023, and now after multi-location testing for 3 seasons, now it has been identified by VIC, which showed overall yield performance of 19% yield superiority over Sama Masuri. And as you can see here, it has got excellent root system architecture along with better panicles and earliness. We have also developed very robust molecular markers so that we can transfer this allele into the different backgrounds. And now with the help of plant breeders from ICAR-IIRR led by Dr. Sai Prasad, now we are transferring this allele and also analyzing the effect of this allele in other backgrounds. And interestingly, so far in 10 diverse backgrounds, we have transferred it, and we found that it showed very superior phenotype. So this was the paper came in very recently, wherein it was will cytokinins underpin the second green revolution. We all know the first green revolution, it happened because of gibberellin hormone, that is SD1. Now the cytokinin, the term is for cytokinin, and it is certainly a hormone that has got a very important role in whole plant biology. In addition to the work that I have shown here that we have done at Indian Institute of Rice Research, my collaborator and also mentor, Dr. Viswanathan, has been working on DST gene at Indian Agriculture Research Institute, that is for drought and salt tolerance. And this particular work was done in MTU 1010. He developed genome-edited line by mutating this DST gene, wherein he created the deletion of two base pair, and this particular deletion led to the plant phenotype that shows very good drought tolerance as well as salinity tolerance. This line also has been identified through VIC this time, and it showed more than 9.6% yield superiority over MTU 1010 under inland salinity, 14% under alkalinity, and also 30% under coastal salinity. So this is performing wonderful under stress conditions.
30 minutes are over.
Yes. So
in
1 minute I will conclude. I am almost done, sir. So these lines have been, these are both the lines are country's first genome-edited lines which have been actually which were deregulated and also identified as variety. This is where now we stand. We have now brought to this level. The only bottleneck is we need to get the access of the technology so that we can make it available to the farmers. We are also trying to couple it with the speed breeding so that we can bring the products as early as possible. So if we connect genome editing is already a very fast technology wherein we can bring the products within 2 to 5 years. But now with speed breeding, we can further accelerate it. I would conclude here. So what we have done in case of Sama Masuri is we could break yield barrier. We have by reducing the duration. We have been not only we have reduced the duration but also increased the yield, and there is no reduction in the tiller numbers. We have identified several climate resilient traits in these lines, particularly the MTU 1010, which shows drought and salinity tolerance. Also the line Kamla that I talked before, it has got excellent nutrient use efficiency. Earliness is going to help and also the lodging resistance. The challenge is for us to get the rights as early as possible, and of course to extend this technology in other crops and in other varieties. We need to develop efficient tissue culture protocols and also transformation protocols. We got blessings and also the guidance time to time from all our seniors and from ICR and also from my teachers. With this, I would like to acknowledge everyone here, my director, Dr. Sundaram, Dr. Viswanathan, Professor Wolf from Germany, Ding Yang, Dr. Nirja, all my team, funding support from NSF, and also all my students. So thank you very much. I'm sorry because of the time I had to rush.
Very good presentation, very informative, and you are seeing the products, and very good. If you visit field, I have seen it. It's a wonderful product they have developed. Thank you, Dr. Satyendra. Next speaker is Dr. Dipal Rai Chaudhary.
PPT. Honorable Professor Mishra sir, co-chairs, and Director Dr. Beg and Dr. Sundaram, dignitaries on the dais and off the dais. Thank you for giving us an opportunity to talk before you, though previous three were high sciences, and we're navigating towards, intellectual property rights. This lecture was supposed to be given by Dr. D.K. Agarwal, our Registrar General, but unfortunately, due to some pressing commitments, he couldn't come. So, pardon me if I cannot make complete justice to it. I would like to talk about the role of Protection of Plant Varieties and Farmers' Rights Authority in protection of plant varieties in India. Many of you know that this was enacted in 2001 and subsequently authority was established in 2005. When it was discussed, it was modeled on eve of plant breeders' rights, but then after the intervention of Professor Swaminathan and some other civil society people, the farmers' rights were included in it. So this act has now, a different platform altogether in the world, because the farmers' varieties can be registered as well as the farmers' rights are to be ensured. So this authority has to ensure that there should be a proper effective system for protection of plant varieties. And then there are several other provisions: breeders' rights, gene fund, compulsory license, benefit sharing, researcher rights, community rights, so on and so forth. It will be very difficult to tell about everything, but in a short, I would like to talk about what are the process of registration. So initially, when it was started in 2001, the government of India identified, about 35 crop species covering cereals, pulses, oilseeds, so on and so forth. And then they established the DOS centers, like at CRRI, at DRR, and so on and so forth, about 59 centers. Then the first 12 guidelines came into being in 2006, notified, and the registration was started in 2007. So subsequently, till date, we have identified about 203 DOSTS guidelines. So once the DOSTS guidelines are notified by Government of India, the registration starts and the applications are invited. So we have an internal team of some technical people and the legal cell. We analyze those, and then if the application is qualified for registration, they will either go for a dust testing, or in case of varieties which are notified under the Seeds Act, they go to a committee. And we conduct the DOS testing at different centers, and then we get the result, we analyze the result, and if we found that the variety is, eligible for registration, it goes into a publication. And unless and until there is an objection on this publication within 90 days from that, then the certificate will be granted. So this is a general procedure. You can go, and all these things are there in our website. Now, when you submit an application, this is one of the major drawbacks that we found, that despite being there in 15 or 16 years, still when the institutes and universities and public or private companies are submitting applications, they made mistakes. And since this application is a legal procedure, it is ensured that you must know what are to be there in this application. So in general, in all applications have to be submitted in Form 1 and has to be along with the technical questionnaire. In case this is an essentially derived variety and the breeder is already here, so there are several essentially derived varieties from ICAR-IIRR and some other companies as well. They submit these applications in Form 2. Now, in case of an applicant from a public sector institute, the director has to be the applicant, and then there are several other documents that they have to submit. These are legal documents. Then the seeds have to be there along with the fees. I will share my this presentation with DRR, and it can be circulated to all of them. We also had an interaction with the rice breeders few months back at DRR during their establishment foundation day. But there are several mistakes even still when we when you get these applications. Only this morning, someone from CRI was asking whether they have to submit all 62 characters or not. And fortunately or unfortunately, she is our nodal officer as well. And she said the breeders do not have all those characters as on date. So this is a problem. So you have to comply with all these requirements in the legislation. Now there are two types of varieties which can be registered. One is a new variety and another is an extant variety. Remember, India is the only country where varieties under extant category can be registered. No other countries in the world where extant varieties can be registered. They all allow only new varieties. And new varieties is something different from our concept in patent. The new varieties are that if you apply today, we receive an application today, and today is 28th of April, so the variety has not to be sold in India between 29th of April 2024 to 28th of April 2025. It is applied more, if it is sold beyond more than 365 days, it comes under extant category. And under extant category, there are three subcategories. The varieties which are notified under the Seeds Act, the varieties of common knowledge, and farmers varieties. And remember, this is why the government of India at that moment of time take a leverage from signing WTO that we will allow our extant varieties to be registered, because our farmers are maintaining these traditional varieties since generations. There are a lot of varieties which are still in the public domain but not yet registered. So we have given a leverage that you can still apply these and get a protection. So, and essentially derived varieties, but can only be when there is an initial variety is already registered. It can be a transgenic one, mutant, black cross derivative, or a somaclonal variant, or even a gene edited, which the some of the scientists were telling that they are in the pipeline. We have established about 135 dust centers. In rice, there are about 8 dust centers. I have shown you the map of India where only 3 centers are there. IRI Karnal, at IIRR Hyderabad, and TNU. Apart from there are CRRI, IGKV, and at the northeast we have Assam Agriculture University, Manipur, and Nagaland. So in all 203 crops, we have established our dust centers who have developed the DUS test guidelines and where the DUS test is being conducted. As you know, I have tried, when you submit an application, you have to submit the descriptor for all 62 in rice. And there are other descriptors in some other crops. When we send the seeds, the varieties will be evaluated stage-wise. Okay, and starting from seed to seed, there are about 62 traits which are to be evaluated at least at 2 locations. And Indian Institute of Rice Research, earlier it was DRR, he is the nodal center. As I said, so these traits we require in the application as well. So many times they are not able to give those traits, but definitely when you want to submit an application, get back to your breeder and get all those characters over here as well. Then every variety has to be distinct, uniform, and stable. Now, what does it mean by a distinctness? How do you establish a distinctness? So, when we get an application, we go for the comparison of the grouping traits. There are 8 grouping traits in rice. So we have a database of about 1,000 odd varieties, and we compare them for 8 grouping traits, and then also other essential traits are compared. So then the centers grow these morphologically similar varieties and reference varieties in at least 2 locations for each varieties. And then the Act says the candidate variety should be distinct by at least 1 essential character. And I have trying to give 2 characters over here which are essential. Based on leaf sheath color, you see one is the green, another is the purple. So these are 2 distinctly different varieties, and similarly spikelet density or pubescence of lemma. There are of course 4 traits, but I have given an example of 2 traits. So there are about 29 essential traits in all 62 descriptors for which the candidate variety has to be different, having a different expression from the reference varieties or from other varieties of common knowledge. Now then, a variety shall also be uniform and stable, primarily because when you register a variety, when you develop a variety, you are developing it for production at the farmer's level. The variety has to show an uniformity in its expression, and it has to be stable over generations. So this is how do we do that? We grow these varieties over generations as well as at different locations. So that we find it out whether the expressions of the traits are stable and the essential characteristics should remain unchanged after repeated propagation. Now, once this is done, we found it out after statistical analysis, the variety is registrable. Then we submit and publish the passport data in our plant variety journal, which is there in published in first working day of every month. So I urge all the breeders or scientists from public and private that you have to be very vigilant, because India is such a big, country where there are millions of rice farmers, and you do not know there are small growers. Probably in Hyderabad, there are about 200 plus companies are already registered. So you do not know whose variety actually they are applying and whose variety that your variety are being developed and marketed by them or not. So the salient passport data is published in the journal along with all the traits, photos over there. And once it passes 90 days and gets no objection, we give the certificate. And the certificate is valid for 15 years for seed propagated crop or for horticultural crops like mango, jam, some other which are vegetatively or asexually reproduced, they are valid for 18 years. Of course, after initial 6 years, you would like to know that whether you will continue with the registration, whether the variety does have its economic potential still, then you have to renew that variety by paying a renewal fee, or you can also say that my variety exceeds its economic potential. In case of extant varieties notified under the Seeds Act, as on date, the provision are that the variety will be for 15 years from the date of notification. That means if a variety is notified in the Gazette of India in 2020, but you have applied in 2025, the 15 years will be counted from 2020, from the day it was notified under the Seeds Act. Now, some number crunching. This is what the rice applications we receive from public and private applicants till date. You see, there are two categories, public and private, and then we have subcategories under them, the new VCK, extant, and EDVs. You can see that there is a biasness of applications submitted in different categories. So, in case of private, the more number of applications are in newer categories, whereas in case of private, more number of applications are in new categories, whereas in public, the more number of applications are coming into extant notified categories.
Rajesh sir, time is over. You may conclude.
I'll quickly go through that. My point is that requesting this platform to all the breeders that you may please also submit the applications under notified categories. There are also few of the applications. You can see there are about 60 odd applications of parental lines. The parental lines also can be registered under the PPVFR Act, and you see there are more number of private companies have applied as compared to the private public sector. There are important provisions, sir. Section 24(5), which is the provisional protection. Section 26 is benefit sharing, breeders' rights, researcher rights, convention country, and infringements. So how the world benefit from plant variety protection? You can see Dr. Kesing sir is also here. Their varieties are being licensed to many companies, and on the IP day on last Saturday, we have given license to about 17 small companies. Of those are from bred by Indian Institute of Wheat and Barley Research. So this is one of the important aspects that they do. What is expected to be done? It is the last slide, sir. So you see that most of the dust traits are morphology based, but now the world has gone beyond that. We have seen couple of presentations on high science. So we have also received applications on herbicide tolerant, which are non-GM mutant of these alleles. So these are not dust traits. So we are also revising the DOS guidelines, sir. So my request to Director ICAR-IIRR to look into whether these characters can also be included. Of course, there are issues with different ecotypes and the methodologies that they may not be available all across all centers, but there is a possibility. So revision is due, sir. So I would like to request that your people were literally busy in AICRP, but beyond that, please submit the revised guidance and then we can have a meeting and complete. One important task that we would like to give it to DRR is that the document, index and catalog notified, released and neglect registered varieties as per DOS. This has been done in 2017 last time, sir. So we would like to, do it at DRR this time. Once you register, you can license it. You also have to pay annual fee and be vigilant on your rights. India has a huge number of diversity, and there are a lot of land races available which are still not registered, and many people are using those. So you have to mentor farmers and farming communities for registration of land races and farmers varieties. Thank you. I hope.
Family, it's a very good presentation, and I think all of you must be refreshed. This is the unavoidable, compulsory things we have to follow it. Thank you very much for the time. Next presentation is from Director MBPGR, and I think Goel sir will come. Vishnu Goel. Huh? Respected chairman, sir, co-chairman, sir, conveners, rapporteurs of the session, dignitaries of the dais, fellow participants. Our directors are supposed to present the here, but due to some pressing need, I have been directed to present the NVPGR assigned activity, conservation and utilization of rice genetic resources. So there are different activities which we have to coordinate through NVPGR, including the exploration, conservation, evaluation, and germplasm exchange, and some of the extent the genomic resources work regarding DNA fingerprinting and others. NVPGR is taking up the activities with its regional stations, and in particular case of rice, our Odisha and Ranchi centers are more concerned with the rice activities regarding some explorations and the germplasm collection, and particularly evaluation at their field locations in collaboration with the CRRI Cuttack. So these are the activities: germplasm acquisition, characterization, conservation. So based on the minimal descriptors in different crops, so minimal descriptors are also available in rice. So characterization part, trait specific evaluation part, biochemical analysis, and further on the conservation of the resources, rice resources, genetic resources in our gene bank. So all the activities are being taken care by the NBPGR. Regarding the status of the gene bank, so total collection is 4.7 lakhs, nearly germplasm accessions are available in the National Gene Bank, out of which nearly 1.18 lakhs are for the paddy collection. Cereals is the major group with 1.77 lakhs of the germplasm accessions. Side by side, some wild species are also available. NVPGR is majorly involved in the genetic stock registration for the PGRC, Plant Germplasm Registration Committee, and we are also assigning IC number for the proposals before the CBRC to fulfill the guidelines. So in 2024, total 95 varietal release proposals were received. These were the maximum in any crop, and for 42 CBRC and 53 SBRC committees, and IC allotment was done. While the overall collection is 1.18 lakh in the NVPGR gene bank, majority of the connections are the indigenous. Land races are the main component, and different state-wise what I could compile with the help of Dr. Suman Roy. Nearly 55,000 accessions state-wise are available. So these are the basically local collections. Every year we are also receiving indents
received by the research centers through ERI germplasm is coming and distributed to the coordinated centers. So some promising accessions reported back, we also keep them in our gene bank for the future utilization. So regarding the wild species, as I mentioned, 17 wild species seeds are available. Some Horizon, Ivara, Rufi, Pogon, Spontanea, Alterna, and the list is there. So these the 17 species seeds are available in the National Gene Bank. But the issue is that we cannot honor all the time the indents, because always we are supplying, so that there are certain guidelines to retain the seed in the National Gene Bank. So my earnest request is that while we are supplying germplasm to our cooperators or some fellows, so we also expect in some cases that the pure seed should also come back to us, so that we can meet out the future demands. Because in self-pollinated crops we are keeping 2000 seed in the National Gene Bank, and number is day by day increasing. In all the case, no multiplication can be take place. So in this regard, the help from this group we can receive. As I mentioned, our Odisha and Ranchi center is involved largely with the rice activities. So time to time explorations are conducted for the collection of the wild and weedy relatives of the rice, and some 154 accessions of Horizon, Nivara, Rufi, Pogon, and Sativa, Spontanea, and likewise were collected. After the collections, here the role of taxonomist is also important. Dr. Mridul also discussed today morning that our Dr. Pani is highly expertise in the issues. But from this RISE group, we can also get the support for some taxonomic identification of the wild species. Day by day, that is gradually decreasing in the system, that the good taxonomists are required. As I mentioned, sir, regarding the germplasm registration, to date from 1996 to 2025, 2,325 germplasm accessions of different crops have been registered. In particular case of rice, 247 accessions have been registered with the ICR NBPGR trait specific germplasm, and largely 89 germplasm were for the biotic stress resistance, including blast resistance, BLB, sheath blight, and other traits. During the recent years, past 6 years, we also analyzed. So new traits now are coming for the input use efficiencies and particularly abiotic stresses. Earlier, the biotic stress character was the major character for the registration. I have compiled some of the genetic stocks registered during the last year 2024, and the situation is very win-win that I could see the introgression from the wild relatives or like Oryza glabrima, Nevara, Rupi Pogon, particularly from this IIRR for BPH resistance and white compatible restorers. As I mentioned, sir, during past 6 years, 112 genotypes were registered with the NVPGR, and number of traits were many. Since the beginning, only the 135 genotypes were registered, but now the number has gone up. So this is the very good situation. But also we have to be very careful while submitting, because as per our guidelines, during the meeting under the chairmanship of Honorable DDG sir, strictly it has been asked what is the value of this germplasm. Already such kind of trait is available, and what is the academic value, what is the commercial value, what is the potential value of this germplasm. So earlier hard applications were invited, but now past few years the online system has been generated for germplasm registration information system. All the available guidelines are also available there, and it is the user friendly type. Drop down menus are available for the friendly affairs of the registration partners. One of the activities is to compile the inventory of the registered crop germplasm. It is very important activity, and over the years since 1906 we are doing.
and many compendiums have been registered, and hopefully all are available online, or recent one are available in PDF form. This was the for registration germplasm supply. It is the mandate of the National Gene Bank, importing and providing to the cooperators. So 12,602 germplasm accessions were supplied by the NVPGR after taking care of due quarantine facilities during 2024. Some of the agencies imported only one accession, while others were taking the many number of the genotypes. I have also mentioned here ICR NVPGR New Delhi. 4,263 germplasm accessions were also provided, because the breeders are also mandated in evaluation division to do activities. Once the germplasm is out from the LTS, so it is in the inventory that we have taken and we have supplied to someone. Earlier, sir, there were questions that we applied for germplasm, but we couldn't get it. So now this system has been gone online. Guidelines are available. Services and fee drop-down menu is available on the NVPGR website. So what are the guidelines? So whether it is a NARES partner, it is the SAU, it is the private organization, all the guidelines are available, and we honor the intents through this domestic supply system. Regarding import, 49% of the import were from the public sector, while majority of the import is from the private sector. And different countries, we are sending the request, while one key commitment is there. If some person is asking or want to import a germplasm from any of the country, so the consent of the provider is also required. If the consent of the provider is available with the request, then it becomes easy that the party is ready to share the germplasm. So this is the list graphical presentation of some of the countries rice accession imported during 2020 to 2024, sir. This is again germplasm exchange and quarantine information system. So means to reduce manual interventions and to trace back your application. Many modules have been developed. ICR NVPGR is also involved with NRRI Cuttack and IGKB Raipur for germplasm characterization under CRP-AB. So some of the genotypes we are providing to our centers, and the seeds, some of the data we are taking back and identifying promising genotypes, though this is the 1 year data, need further confirmation. This is some of the pictures of color variations received from our NRRI partner, Madam Sangamitra. Some morphological trait accessions were identified for leaf length, leaf width, effective tillers, panicle length, and others. Sir, during 2024, earlier I was in Karnal, I joined in 2024 at NVPGR, so rice work was not there. We also started under the guidance of our director, and 8,500 accessions we also planted under DSR conditions, and one student activity was also organized to share the rice knowledge on the rice genetic resources. This is the native basket initiatives were taken care with the Biodiversity International, where somewhere in 2021, 635 local land races from Assam, Chhattisgarh, and other states were evaluated for quality characters, and based on the farmers' preference, local demand, some of the land races were identified, and local farmers, on the basis of the need of the local cultivable land races, selling out their seeds being remunerative to those farmers. So this is the native basket from Assam. After going, just I will take 2 minutes. Core development, it is the activity. So molecular cores were developed from the northeastern core and coastal saline areas. So 701 accessions were categorized in one core and 247 in another core. And this year we also supplied all the material to IIRR Hyderabad for multiplication and keeping back in our seed gen bank. So some partner is asking for, so that is also under process, and we will get back seeds soon from this place. Some To develop a database, some basic studies were also carried out. I think Dr. Rakesh Singh sir take up this activity under the guidance of Dr. A.K. Singh sir and Dr. N.K. Singh ji. And one database has been prepared based on the SNP and SSR markers for the 630 varieties, released varieties, and that is helpful in some case of the means controversies. It can help bit extent. So thank you very much, sir.
Thank you, Dr. Very nice presentation. Interaction will be after the presentation. So the next is a very important speaker. They are Dr. Vikas Kumar Singh from ED, Dr. R.M. Sundaram, our director, and then again it will be followed by Dr. Vikas Kumar Singh and Dr. Nesar Srinivasi.
Good afternoon, everyone. So this is a joint presentation with me and Dr. Sundaram. So Dr. Sundaram will present the first few slides. After that, I will take it over from there. I'm just waiting for him to come. Meanwhile, this first presentation will be the overview what we have done in the 2024 in from all the TA or the work plans. Later on, we have two presentations which we will talk about breeding and trade specific, work, what happens in the region. So that I will present, and the next presentation will be by Paneer. He will give us an update on agronomy side. So total 3 presentations will be there. So meanwhile, Dr. Sundaram joins. Okay. But that is actually flow of presentation, so it will not look good to connect. But meanwhile, I'm waiting for Dr. Sundaram. I'm just setting the stage. So in the ICR work plan where we have worked, it was designed with 5 different thematic areas. And the first thematic area is about how we can develop market-oriented rice varieties. And it was led by Dr. Sai Prasad sir from IIRR and myself from ED side. Thematic area 2 is basically about providing integrated solutions for direct seeded rice. Dr. B.B. Panda from ICR NRRI and Dr. Virendra Kumar from ED. I was waiting for you. I'm just giving the floor to you. Third thematic area is about rice-based health and nutrition, led by Dr. Harita and Dr. Gopal Krishna of IRI, and from ED side, Dr. Nese. Thematic area 4 is about climate resilient rice, Dr. A.K. Nayak and Dr. A. Kumar from NRI, and from our side, Dr. Paneer is here. And integrated capacity building was led by Dr. Sundaram from IRR and Annalyn from ED. Dr. Sundaram, please.
Sorry, in between there was a call from head office, so I had to just go and attend the call. Thank you very much, Dr. Vikas, for setting the stage. So now coming to the progress. Last year when our collaborative work, when I say it's a collaborative work, it's not just a collaboration between IRRI, IARR, CRRI and IRI. All of you are proud partners in it. For just for organization's sake, some conveners and coordinators and then joint deputy coordinators have been identified. Each one of partners from NARES, including SAUs and other organizations, even private companies, are also proud partners in this collaborative network. So a few suggestions were made when last time it was reviewed by ICR. What they wanted is, IRRI together with partners, they were supposed to update the available technologies across thematic areas For example, this can be classified into tools, specifically for breeding related tools, DSR and mechanization related tools, and other natural resource management related tools. For example, as far as the breeding related tools are concerned, we are proud to say that when we wanted to construct a speed breeding facility, IRRI South Asia Regional Center came forward and then immediately offered us all technical and then other assistance in terms of how to design the structure and what are all the materials to be used and all. Similarly, we are also getting outstanding support in terms of genomic services, they have developed several set of tools which we are using. We means I talked about the entire NARES partner in the systems, and they are also providing excellent ICT support also, and they are also working for upscaling of some of the climate resilient varieties and other things. I'm not going to the details of it. If anybody wants, I can share a copy of these proceedings. And we were also asked to map hybrid rice resources and strengthen two-line breeding program. In addition to supply of germplasm to IRR and other NARS partners, IRRI has also given some advanced lines which can immediately be evaluated by different equip centers and they can be nominated for trials. And one success story in this particular regard is that IRRI had a very good restorer line. It was used as the restorer parent in our breeding program with our own CMS line. And last year, we are proud to claim that through this partnership, the first or probably the world's first salinity tolerant hybrid was released called DRRH4, and it has got medium center category. Like that, there have been so much of success story, and already these lines have been multiplied at IARR, the parental lines, and it will shortly be distributed for all our partners. Just send us a request, we'll immediately send it to you because we have got sufficient quantity of seeds. And same is true with regard to nutritional improvement also. We were requested to share high oil content rice lines with IARR and other partners. So with regard to this thing, already discussions are going on, and from 2026 onwards, this particular activity will go in full-fledged this thing. And the fourth suggestion was infuse iron and zinc rich advanced lines through breeding programs. Together, we are in the process of developing neurosygenic lines through haplotype-based breeding strategy, and lines are in various stages of testing. And already one variety was released last year called Spurti, which was a collaborative effort principally driven by our rice research station Gangavati along with IRR and IRRI, and other lines are also in the pipeline. And the fifth suggestion was incorporate water use efficiency and nitrogen use efficiency rice lines into the National Improvement Program. Already, this is part of two of our breeding platforms or pipelines, and this work is already going on. And finally, they also wanted the team to identify multiple disease-resistant varieties or genotypes. A large set of lines have already come from IRRI. This was developed through a collaboration, and they are there in various breeding programs. And we also got access to those lines. And I'm also happy to claim that one of the breeding lines sent by IRRI, which has got introgression from Boreza officinalis, we were successfully, together with IRRI, we were able to tag, map, and clone the candidate gene. And the new gene, which is a dominant resistant gene, has been named as XA48. And we have got neurosygenic lines of Samba Masuri with this particular gene. Then, breeding work under this particular platform is being done through various maturity-based pipelines. And the pipelines are also classified based on not only maturity, but also grain type, targeted towards different ecologies. I'm not going to name all these things. So through these pipelines, already we are evaluated through a combined team consisting of not only the major ICR centers, but also our proud NARES partners. We have conducted evaluation of these lines, and some promising lines have already, as I mentioned a few examples, have already been released through CVRC or SVRC, and the list is already growing. And another important initiative is that, considering that coastal salinity or inland salinity is a major problem, we wanted to initiate a specific breeding program targeting salinity. In this regard, already the partnership has already been crystallized, and we already identified about 10 testing sites, and we are going to shortly apply for a joint funding from national and international donors. And We also formed a regional breeding pipelines and then joint crossing block. This is a work in progress and then it will continue. Finally, with regard to capacity building, it has been a very good set of activities which we have done. Together with IRRI, IRR, CRR, and CRRA, and then IRI, we conducted regular capacity development programs wherein all our newly joined AGRM cooperators, they were all trained about what is the AGRM system, what are all the requirements of the system, how to carry out the trials, and also how to analyze the data, and also how to feed the data online. All these aspects were trained. And we also, in addition to that, some specific subject-specific trainings were also offered, either in online or offline mode or hybrid mode, in terms of training on biometrics. We also had our advancement meet and hybridized policy workshop. This was a huge success story. Last year it happened. And we also conducted virtual training on AGRM experiments through online. And we also had an offline training course, which was, I would consider it's one of the most successful training courses on fundamentals of genomic prediction and data-driven crop breeding, a topic aptly highlighted by our beloved ADG, Sir Dr. Pradhan. And we have also, through this program, hosted many Indian scholars at IRRI. And we have also, in collaboration with several state agriculture universities, some of the students have been visiting IRRI for a month-long or longer period exposure visit at some of the advanced facilities at IRRI. And we also initiated a ICR Student Ready program, which is being operated at Center for Excellence in Grain and Cooking Quality at ICAR Varanasi. And together as a team, we also developed several micro-learning modules and other tools. I would like to stop now. I request, of course, one more is also there. These are all some snapshots of different online and then offline training courses conducted combinedly with our AGRM partners and also with IRRI. Thank you, Dr. Vikas. I think you need to continue.
Thank you, Dr. Sundaram. Yeah, thank you, Dr. Sundaram, for again setting the stage. So we'll take you more about what we have done in 2024. As you know that we have established this not only speed breeding center at ICAR, but we publish a protocol, what we call as a speed flower protocol. And based on the suggestions again from DDG Crop Science last time when he visited, that this is the time to start working on wild rices. So we start, working closely with IRR to collect and to develop a protocols, what we name as a speed wild. And idea is to develop a flowering protocols and how we can develop, populations which we can share further. The new development which is coming again from the same area ICR work plan, and I remember the suggestion provided by Dr. A.K. Singh sir, that while you people are working on several haplotypes, why we cannot develop a new chip which will be useful for genotyping. So Pallavi and team along with colleagues from IIRR, IRI and CRRI, we discussed and we finalized this new chip. Still numbers are being fixed. It's called the 4K haplochip. We are targeting more than 2,200 genes, and this will be capturing all the traits, whatever we work right now or in the future for many of the applications. At the same time, Dr. Suresh sitting here, he is developing another chip. It's called the 4K chip, mainly for genomic selection applications. And these two chips will be available very soon for breeding programs or trait discovery programs. In terms of hybrid, a lot of discussion happens that what we are doing, we are again working very closely with the ICR, and not only we do have a discussions, we have provided a lot of genetic materials, and ICR also joined TLSG membership. So this year we are planning to have this material released. So later on we can have much better hybrids. Also, we have shared additional sets of restorers with resistance to multiple stresses and false spot as well. While we develop the products, it is also very much important to do on-farm validation. Again, Swati is here, and we did large scale, mini kit trials of release varieties, many OFTs in of all the new genetics in many states. Generally, we discuss again in the advancement meeting what we jointly coordinate. We get the nominations, and we used to share all the results of these. While we do this, we also discuss that how we can accelerate the adoptions of these variety. We work with the private sector partners as a seed accelerator workshop. And as you can see the numbers, we are working really very closely with all the KVKs, FPs. While we do this, we also did a lot of training programs to build the capacity. Moving to the next specific direct seeded thematic area, where breeding and agronomy team work together. We are not only implementing the stage 1 and stage 2 trials, more than 40 experimental locations in India. We have also designed a new plot planter, which will be very useful to keep the trials ready. And as you know that we have a state-of-art phenotyping facility at South Asia of Hyderabad. We grow all our material there, and we generate all the data of these new genetics. While doing this, along with our national partners, we identify the best products which shall be useful for different trials, and we nominate them. At the same time, we also have a discussion that new upland pipelines need to be required to fill the gaps in the breeding program. As we develop a product, we also do a lot of innovations. Now we have started 1 year breeding cycles. If we require more details, we will present more results in tomorrow's advanced meeting. As I have explained that at one side we are developing a new genetics, but we are also evaluating all available inbreds and hybrids. And the key findings are that definitely hybrids are doing much better in comparison to inbreds, which are also very much reflected in the IVT DSR trial, what recently data released. Not only about yield, we are also talking about how we can optimize the water schedules. All this DSR and agronomy trials. When Paneer will come, he will explain in much detail. I'm just giving you the overview. We have identified what should be the minimum kilopascal water capacity where we can have same yield without yield penalty. We have did this trial at PIU Ludhiana and the Karnal. And we are talking about HT technology. Last year, the one thing what happens, we along with our all ICR partnership, we did a workshop at Varanasi to develop a stewardship for HT rice. That was a very important outcome of this trial. We are also developing a protocol for drone-based spraying and weed controls, and also recording the data of the GHG emissions. Without mechanizations, DSR will not be very much successful, and we have done a lot of mechanizations based trials in Odisha, and we have observed this time it is having 8% higher yield in a direct seeded conditions, and 16% more yield than the bushing methods. As I have already mentioned, hybrids are doing well in comparison to inbreds. We are involving several farmers for participatory demonstrations, so DSR technology can, be reaching to a large number of stakeholders. We are also providing service providers, training, so we can have much more better services available in terms of machineries and support. Now we are moving to TA3, which are also very important and critical TA, where NESE, along with our national program partners, are working to develop low GI rice varieties. And based on his recent, publications and the outcome, he do have now a genetics combined with high protein and low GI rices. One of the genetics we have identified together, which is of low GI in nature. And this line was nominated last time in the biofortification trial. We'll see the performance of this line, and accordingly we will prioritize for possible release. Okay, thank you. Thank you so much. This is again a research work of NESE where he identified important locus on chromosome 2, which controls the low glycemic index. And finally, we have also validated this locus via genome editing, and we found this trait or this gene is working well in the background. Moving to the next TA, this is a TA4. This is again led by Paneer, and here our objective was that how we can combine several traits to develop climate resilient varieties. And we developed, a pipeline while working together with many national programs, from where we can identify the traits to the products, I will cover this slide in very much detail in the next, presentations, that how we have done and what are the things available with us. Moving next, and this is again a very important activity where BAN is working closely with again Dr. Sundaram and the list of pathologists. We have collected 184 BLB samples across the regions. We have genotyped. We found these three populations are very much prevalent, and still all the four genes are very much effective for controlling bacterial blight. The right side is again a very important flagship project where we are again working closely with the national programs. And the idea is that how we can really characterize all the emerging diseases, brown spot sheath blight, false spot, and PPH. We do have a large number of collections. Currently, all these uh, uh, all these pathogens we are isolating pure isolates, and we are subjecting for the WGRS. The idea is to did a pathogen, diversity. At the same time, we can map the trait of interest. Yesterday, when DK Yadav sir was explaining about the applications and machine learning tools are required in the area of agriculture, at IRRI we do have a very fantastic tools developed. One of the called is the Deep Map. Deep Map is a artificial intelligence based tool, again developed regionally, led by Pallavi, where you can do all your calculation of breeding values, which will be helpful for the breeding program. Next is again a lot of experiments happening for the greenhouse gas emissions and how these are helpful under AWD and transplanted conditions. I'm living where I believe that Paneer will explain in details. Also, a lot of experiments we are doing for improving resilience in rice productions. That how biochar will be helpful in increasing soil organic carbon. I believe these are the trainings associated with the topic what we have talked about, and I still believe that again, Paneer, you need to present in much more detail. So this is a just overview what we have done under ICAR work plan. So,
Thank you, Dr. Vikas. Wonderful presentation. Control time. Dr. Sundaram, you want to say anything?
yes, I will take only 5 minutes. I will just show you the major results. As you know, the second presentation was about updates on traits and breeding side. This is a slide where you can see the journey of molecular breeding research in India. A lot of fantastic products we have developed over the year, from bacterial blight to blast, bacterial blight blast combined, submergence, QDTY, and now very recent products we have seen with herbicide tolerance. Very beautiful story, and you will not find any such a story in any of the crop across the world. At the same time, when we talk about, we do have a now next generation of genetics where we have combined not only one or two traits, but many traits like BLB, blast, gallmage, BPH, drought, submergence, cold in one genetic background. Not only one, 12 different genetic background. This work we have done in close collaboration with IRR, IGKV, Raipur, TNU, and 177 lines, fixed lines, we have distributed to IRR, CRRI, and IRI for possible use in the breeding programs. We talked a lot about direct seeded rice. We also combined several important genes and QTLs in two different backgrounds, one in MTU 1010, another is IR 91648. And these are not NILs, these are the pre-breeding lines. We have combined and developed several forward breeding and MABC lines, shared top 48 DSR lines to again with the three ICR institutions for further applications and use in the breeding program. These are the new sets of native trait lines we received from Damien. And you can see these all are actually NILs in the background of IRRI 154, which carry different genes from blast to bacterial blight, fragrance, cold, and salinity. These all are we have shared with 13 NARS partners, and they have evaluated are being utilized in the breeding program. Now, as Dr. Pradhan was explaining about haplotype-based breeding, will take again few minutes to explain. And this, slide presented by Satendra in the morning, when he was talking about GN1A discovery, this is a very again discovered long back, but is still very few of the institutions are able to transfer this into their breeding program. In 2018, we are able to transfer because we are able to get original Doran Habataki into the breeding program. So the only problem what I have seen that as in one side you see the very successful example of molecular breeding happening in India and in not only rice, many crops, but we do not have access of original donors which can be utilized in the breeding program. So we came up with a concept where we call as a haplotype based breeding, where we are not only able to identify the candidate genes, because in the case of rice, more than 4,500 genes already cloned or characterized. We do have a 3K panel. If we are able to phenotype, if you are not having a 3K panel, we can take another panel. We can identify the trait of interest using all these tools in our locations here. And we have developed again a fantastic pipelines, what we call as a graphical user interface based pipelines, which can help you to find genes to haplotypes for your donor program. As I have already explained, we do have now new sets of genes. We do have a speed breeding technology. We have developed many haplotype based, NILs in collaboration with the many institutes. And after doing, a lot of trait phenotypings, many of the lines nominated in ACRIC for, testing for possible release. I will again not read for the product concepts, but we know that for developing any product, you require full sets of, traits by which your plant or variety will stand in the farmer's field. So what we did, and you've seen this slide, where we have discovered large number of haplotypes of already known genes, but that time we do not have a donors in hand. But what we did, as as I was explaining, NASE identified important genes. For low GI, we can identify, the alternate or best haplotype, just phenotyping the 3K panel here itself. I have already explained this slide. Now this is an example of haploanils. When we have find important genes for drought tolerance, we not only identified the superior haplotype for drought tolerance, we transferred in 3 different background, Naveen DRR Dhan 44 and RNR 15048 to see the performance of this line. And you can see that every background, it works very well, where we have increased grain yield under drought from 7% to 33%. This is a new set of genetic materials we have developed locally while working with several of our national partners. IRR is one of our close partners. We developed 69 new generation of NILs. I will not read the these genes. These all are very important genes. As you have seen the impact of one gene when Satyendra was talking about GN1A. This is all a very important and critical genes for increasing grain yield, for increasing grain zinc, and we have transferred in 9 background. Every haplotype of these genes you will find in 3 different genetic backgrounds, and these are now we are evaluating closely, which are will be the promising lines we will share further for further use in the breeding program. So 2-3 minutes, I will just close it. Now this is a research where Lavi and group is working to tackle few of the problems which still we are not able to handle. The first one is that whenever we do have a gene in hand, and you transfer into any background, sometimes it give you the same expression, sometimes it will not give you the expression. But what will happen when you have a many genes where you have to incorporate? What will happen after several years of research? You are able to know that this gene or this QTL's interactions are not working well. So HapPredict is a new artificial intelligence based tool we have developed. As Satyendra, put a very important slide that 60-70% of the time and efforts required when you're setting your experiments. So HapPredict will tell you which combinations of genes and QTLs in which background we need to start a crossing program. Need not to take all the backgrounds and all the gene combinations while you are doing. So HapPredict will tell you exactly same. It will give you a very good results, and this tool can also be useful in genome editing research, because currently we are handling 1 or 2 traits. In future you have to handle many traits. So we need to know which combinations of genes and backgrounds need to work actually. The deep map I have already explained. This is a tool where you can find out best combinations of parental lines. It will help you to estimate the GBVs. This is the most important, a tool, what we call as a speed map, because still out of 3K panel, what we have at IRRI, 10,000 germ plants, what we have seen, are being phenotyped at various places. we are not in a situation to phenotype for all the traits. So sometimes we create a smaller panel of 300 lines. So speed map, what it will do, if you are able to generate very high quality precision data of a smaller subset, 200 to 300 lines, it will help you to predict the every possible combinations of trait of interest and artificial intelligence based, all the traits we can utilize to predict or to map into the genome wide association mapping. So we have used this technology in many of the traits, and we are about to release this pipeline in the paper. This is one of the drawback what you will see, that sometimes as a breeder we are very good in phenotyping and genotyping, but we are very poor in analyzing the data because it is very difficult for us to run all the tools. So we have developed two tools. One is ET-GWAS and HAP-GUI. ET-GWAS is helping to extreme trait-based GWAS. You have to just upload your phenotyping data. If you are working on 3K, it will automatically extract all the genotyping data of 3K genome, and it will give you the fantastic results. The right side tool is a HAP-GUI, where it will analyze all your phenotyping data. It will analyze, the 3K data. It will do the GWAS from 5 different models. It will help you in identifications of haplotypic diversity of a genes. Then it will also help you to find out superior haplotypes. So this is a one and all package where you can have a donors in hand for your breeding program. So with this, I'm just stopping here. And the main important thing is that how it is actually deploying different technologies to not only develop products, but to map different genes, how we are combining, genomic selections with the haplotypes, how new array, as I have already explained, 2 new array will be there for breeders, which we can utilize in the breeding programs, which will be helpful for further, to develop much better products. So thank you very much. Thank you.
Updates on agronomy research by Dr. Paneer Selvam.
Thank you, sir. So I'm actually presenting the combined two teams from DSR as well as climate resilience. Dr. Virendra Kumar is leading DSR, and I'm just going to talk about the topic I mentioned here. And we have been actually, IRRI have been working with the partners, both private and public partners, on DSR and developing agronomy, best agronomy. Around 20 partners across 14 states, we have been working on different intervention or practices we have listed here. We can see on the right side, including varietal evaluation and other agronomy practices also. So when we look at that, here I am actually presenting for rice grain yield and net profit across 4 locations. And we know this is particularly this is for dry DSR, and the performance of dry DSR is depends upon the soil type and management. We can see here that the yield is almost similar across the states, except in Tamil Nadu. Tamil Nadu, we can see here that DSR yield is better, that is like a 0.5 times. But the most important that dry DSR is able to produce more income compared to PTR. We can see here around more than $200. And also we have doing this varietal evaluation more suitable for a dry DSR. We have mentioned here partners, and these are the inbred and hybrids performed better across these different locations. The performance of varieties and hybrids really depend on the different location, also duration. In nutshell, actually, as Vikas already said, hybrids are performing better in a dry DSR compared to inbred, and also PR126 also in case of inbred actually performed better in Punjab. So this is for West Bengal for Boro season. Similar, the hybrids are performing better both in weed free as well as partial weed condition, because of hybrids have more vigor and weed competitiveness. so the yield will be higher under hybrids. And also because we have been talking more about DSR water saving, but it also depends upon the location soil type. So we able to measure water saving and water productivity across these 4 states. And we can see here that around 30% water saving in case of Odisha and UP, and around 50% water saving in case of Tamil Nadu. And also we are trying to optimize irrigation scheduling for DSR, because there is not really much actually evidence when to irrigate for DSR field. So we are using tensiometer reading, and also we have actually come up with a different kilopascal, which up to which level we can actually go for irrigation. We can see here that 20 kilopascal is more safe. There is no yield reduction. We can save water around 25%, and we can also go up to 30 to 40 kilopascal. Water saving is there around 40%, but there is a yield penalty in case of PR 126. But DRR 42 is able to withstand a more tolerant without yield penalties. This is from PIU. The same case, the same experiment we have done in the CSRI also. The results are almost similar. And this experiment we have been doing in ICAR last 6 years. Actually, we are trying to impose in the, drip irrigation, both subsurface irrigation and subsurface drip as well as surface drip. And we can see here water productivity in case of DSR subsurface drip irrigation on water productivity is more than 3 times compared to PTR flood irrigation, and the yield is almost similar. And so we can, but when we go with the DSR in flood condition, water saving is less, and the yield penalty is observed compared to PTR. And the last year we initiated one experiment in TNAU. Actually, because dry DSR provides the opportunity for intercropping because of there is no puddling and flooding. And we actually integrated Sespania in this dry DSR. And also we also introduced the fertilizer deplacement, comparing with the 100% RDN for broadcasted method. And also compared to PTR. And also when we go with the nitrogen application, that is even 75% recommended dose of nitrogen through 50% around fertilizer deplacement, and then remaining 25% through broadcasting in chemical initiation. We can see yield is almost more than 1 ton compared to N2, that is the state recommended fertilizer recommendation. And also this nutrient use efficiency also higher in this N5 and N6, that is the 75% RDN through fertilizer displacement. That means we can save around 25% of nitrogen without any yield penalty. Even we can increase the yield compared to N2. So this is already Vikas talked about this slide. I'm not going to repeat. I'm just moving on that. We also actually doing herbicide tolerant, actually some experiment with the NRRI and IIRR also. And we can see that the W3 and W4, that is the emulsifier, we tried, and the yield is almost the same compared to W1, W2, where those herbicide treatments are popular and better. And for both varieties. And also this is also from ICAR-IIRR. We can see here this even one post-emergent herbicide, that is the WM2 is almost similar to pre-emergent herbicide followed by post-emergent herbicide. But we can see the varietal actually makes a lot of difference in performance of yield. And also we have with the IRRI, with the IAI, actually we also doing greenhouse gas experiment, and also couple of PhD students working with the ISAR. On this aspect, we can see, we know that AWD practices mainly developed for water saving, but now we mostly focus on this technology is reducing the greenhouse gas emission. We can see around 54%. And there is a concern also that people talk nitrogen oxide is maybe increase under AWD, but we are not actually when we measure that it's almost similar. So more important that global warming potential is reduced under AWD by 36% compared to PTR. And in addition to that, we also try to impose different soil amendments and also changing some practices instead of urea top dress, and we also gone with like ammonium sulfate. So how is this is improving or reducing the methane emission? We can see, we know that already AWD reduced the methane emission, and there is not much difference in case of nitrogen oxide emission. But when we look at these different soil amendments and our nitrogen application through different source of nitrogen like ammonium sulfate is T5. There is a huge difference in reducing methane emission compared to urea application as a top dress, that is a T1. And also when we combine with the biochar as well as phosphogypsum is also able to reduce methane emission, but there is a increase in case of methane when we actually incorporate rice straw compared to T1, that is the recommended dose of fertilizers. And we know that DSR will not actually go everywhere, will not adapt everywhere because of due to many constraints, including soil type. So we able to actually map suitable area for DSR. Example for Odisha and also for Haryana, we can see here. And Odisha around 0.5 million hectares are suitable for mechanized DSR. So mostly I'm talking DSR, mostly mechanized DSR. And also under one project we have IRRI in collaboration with the Department of Agriculture. We also done cluster large scale cluster demonstration. Here cluster means not 1 hectare, 2 hectare. It is around 25 to 50 hectares. We actually combined many farmers in that, and we actually promote mechanized DSR, and it is really able to, the farmers is able to get more yield and also income because of reduced cost of cultivation. In the same case also in Bihar and Chhattisgarh and also UP, and we have done around 25,000 field days and exposure visit and other dissemination technology on this aspect we can see here, and around 475 agriculture events also we have conducted, and the participants are around 24% of the participants are female. So catalyzing the mechanized DSR more important because individual farmers cannot have access, cannot have the machinery or seed drill to go for seed, but we mostly promote the DSR through the service provision model. We can see that in Odisha around 480 service providers are we created, and mostly all those the area I was talking mostly promoted through service provision model. In Bihar, we know that zero tillage wheat are actually more area, but we try to convert some of the city wheat service providers for the DSR also in Kharif season. So this is all from my side, and thank you.
Thank you very much. Very good explanation. Thank you. So it's now open for a few questions, interaction. Yeah, we all are very hungry. You can differ.
Yeah.
From whom you want to.
Okay. Disease and insect reaction of genome edited lines. What is that status?
Dr. Deepak Sharma ji wants to know whether the disease and pest reaction of genome edited line is it same as Samba Masuri or is it different?
Sir, for Samba Masuri, disease reaction is exactly similar to what Samba Masuri had. With respect,
keep the mic closer.
With respect to pest, we got it shows resistant to galmage. We are looking at it why it is showing, but this genome edited line is showing resistant to galmage because It could be due to, I do not know if the cytokinin has got any role to play in the resistance, but otherwise it is showing similar reactions.
Dr. Goel.
Actually, one line was imported from Malaysia via. So, but we should be very cautious about its quarantine, this treatment. So they have done this hot water treatment. So it loses its viability, germination. This is in most of the cases. So what is the answer about that in this issue, sir? In most of the lines, this happens, sir.
Sir, while import is there, so quarantine division takes care of that. Yeah, regarding that, yes, sometime it happens, sir, and further update I will let you know personally because I am not much aware about this issue. Very good question.
Hi, good afternoon, everyone. This is Balaji from Mahindra Agri. My question is to the person who has given the PPFRA presentation. It's well that we are using those characteristics till now to identify the varieties or the registered varieties. I just want to know that how we are going to leverage the present current technologies like S&P technologies to protect our varieties. Since the numbers are growing day by day, I want to know that what are the actions you are going to utilize the S&P technology to protect the varieties. For the PPFRA registration.
People is not there.
Okay.
So what I suggest is that we can have a discussion with him through email, or I can also give his phone. Since sir is not there, we cannot answer on his behalf. So sir, my humble submission is, there are a lot of questions you may have, each one of you, but because of paucity of time, we cannot connect. Okay. Madam, please, we are already delayed for lunch. Food will get cold. Let's hear the chairman and co-chairman's remark. And what I suggest is that if there is any query, if you're not able to interact with the speakers, send those queries to me. I will get the answers for you. Okay. Thank you. Okay. Sorry.
So now may I request my friend Dr. DN Singh to give his remark.
थैंक यू मिस्टर चेयरमैन। सबसे पहले तो आई एम वेरी हैप्पी कि चेयरमैन और मैं दोनों एमएससी एजी में गवर्नमेंट एग्रीकल्चर कॉलेज कानपुर में क्लासमेट रहे। तो 2 साल हम लोगों ने साथ पढ़ा और आज डॉक्टर सुंदरम ने जो हम लोगों को दोनों एक साथ जो है प्लेटफॉर्म पे एज ए चेयरमैन एंड को चेयरमैन का है। तो थैंक यू डॉक्टर सुंदरम सर।
थैंक्स आल्सो ड्यू टू एडीजी सर आल्सो।
अब एडीजी का तो पता नहीं है, लेकिन हम आपको धन्यवाद दे रहे हैं कि 1966-67 में हम लोग दोनों साथ पढ़ते थे और इतने सालों बाद हम लोग फिर साथ मिले हैं एक ही चेयर पे। मैं कुछ बातें कहना चाह रहा हूं। काफी लोग जो भूखे थे वो चले गए बाहर, लेकिन जो लोग हैं उनकी पेशेंस को हम दाद देना चाहते हैं। मैं 2 एग्जाम्पल बताऊंगा। अभी डॉक्टर खुश ने जो है किताब लिखी है, सॉरी मैं हिंदी में अंग्रेजी मिक्स कर दे रहा हूं। डॉक्टर खुश ने अभी एक किताब लिखी है, पिछले साल या 2 साल पहले उन्होंने ओडिसी मतलब अपनी क्या कहते हैं कि यात्रा, और उसमें एक बड़ी इंपॉर्टेंट चीज बताई है कि हाउ ही वाज एबल टू ब्रीड ICAR-IIRR एज ए गालमिज टॉलरेंट वैरायटी, क्योंकि फिलीपींस में गालमिज नहीं होता है। और उसमें उन्होंने लिखा है, डॉक्टर पद्मनाभन वाज द डायरेक्टर एट दैट टाइम एट CRRI and they met somewhere in the Hungary or Poland in one of the symposium, अभी वो हंगरी और पोलैंड में थोड़ा कंफ्यूज कर रहा था, और वहां उन्होंने उनसे रिक्वेस्ट किया कि आई वांट ए डोनर फॉर गॉलमिच। और व्हेन ही केम बैक टू कटक, डॉक्टर जेके राय वॉज दी ब्रीडर एट दैट टाइम, और जेके राय साहब से कहा, फिर क्योंकि मैं जेके राय साहब से कंफर्म किया और मैडम भी हैं, उन्होंने फिर कंफर्म किया। देयर इज द मेल पेरेंट ऑफ द IR36 इज CR94-13, or that is a cross between the PTV18 and PTV21, and this gives not only the resistance to the gall-midge but also the BPH, GLS and other. So the traditional donors, where CRRI was trying to combine the resistance of gall-midge, BPH and other traits from the Kerala, Patambi Kerala, so that was 94-13, and that line was sent to IRI, और डॉक्टर खुश ने वहां क्रॉस करके, and he sent back some of the recombinant lines जो उन्होंने वहां सेलेक्ट किया था टू CRRI, और यहां उन्होंने वो गॉल मिज रेजिस्टेंस स्क्रीन किया, और फिर उन्होंने वहां सेम लाइन को जो है इरी में सेलेक्ट करके, एंड देन ही वाज एबल टू रिलीज द IR36 एज ए गॉल मिज रेजिस्टेंट वैरायटी, जो कि व्हेन आई वाज इन झारखंड, समटाइम दे वर रिकमेंडेड फ्रॉम द स्टेट गवर्नमेंट कि हम लोगों को गॉल मिज के नाते जो है राइस कल्टीवेशन कुछ साल के लिए बंद कर दीजिए। अभी भी IR36 जो वहां ग्रो होती है, बिकॉज इट इज रेजिस्टेंट टू द गॉल मिज। सो द मेजर मैसेज इज कि हम कैसे दूर बैठ करके, या इंडिया में भी डिफरेंट स्टेशंस है, कैसे हम कोलैबोरेट करके, and we can select the multiple disease and pest resistant variety. This is an example which I wanted to say from Dr. Kush's book, he has written as a ODC. Even IR64 is also, many other things are there, but I found this is very interesting. अच्छा, दूसरी बात यह है कि जो 94-13 का पेरेंट है, मेल पेरेंट, CRRI ने क्रॉस नहीं किया। जब उन्होंने पूछा कि when I asked him कि whether this was utilized in the CRI breeding program, उसकी सिस्टर लाइन तो इन्वॉल्व हुई थी, लेकिन that line was not. So this is what I wanted to say, Dr. Saab, how the collaboration, international collaboration or the national collaboration can help in developing the breeding objective and multiple resistant variety. एक और एग्जांपल दूंगा। मैं एक बार आदुतुराई गया था, और आदुतुराई ने CR1009 जो कि रिलीज किया था पुनामनी, नाउ इट्स ए वेरी वाइडली ग्रोन वेराइटी है, उसमें सबमर्जेंस जीन डाल करके CR1009 सब1 हैज बीन डेवलप्ड। इट्स ए वेरी गुड वेराइटी फॉर द कोस्टल एरिया। वहां एक मैडम ब्रीडर थी, आई वेंट टू हर इन द ऑफिस, और फिर मैंने पूछा कि हाउ डिड यू सेलेक्ट दिस CR1009 वेराइटी? तो मैडम ने जो बताया वो इट्स आई ओपनर टू ऑल द साइंटिस्ट हियर। उन्होंने कहा कि एक्रिप से एक ट्रायल आया था, और उस ट्रायल में ये वैरायटी वाज हाई यील्डिंग, हाईएस्ट यील्डिंग, या मतलब टॉप 5 में था, एंड द सेकेंड ईयर दे फर्दर कंफर्म इट थ्रू द इकोनॉमिक ट्रायल एंड दे रिलीज एट पुनामनी CR1009। तो ये जो एक्रिप की मीटिंग हो रही है, दिस इज मेनली ए जर्म प्लान और इम्प्रूव जर्म प्लान। उसका एक्सेस है जो हमारे 45 जो क्या नाम है मेन सेंटर है, या जो वॉलंटरी सेंटर है, यू आर गेटिंग द बेस्ट लाइन फ्रॉम द डिफरेंट नेशनल एंड इंटरनेशनल सोर्सेज, एंड यू हैव द रिस्पॉन्सिबिलिटी ऑफ द इंडिविजुअल ब्रीडिंग स्टेशन। उसमें आपका जो बेस्ट चेक है, you select the better yielding lines from the best check, further retest it and release it for your environment, और फिर उसको आप you give it to the further testing in the ACRI program so that others can also take benefit of it. So that CR1009 is the product of the ACRI testing, and there now the submergent tolerance gene has been incorporated, and this is a very good for cyclone and flood prone area of right from West Bengal up to the Goa. So this is how the improved germ plants from the ACRIP should be utilized for the local breeding program, select the best line irrespective from where it comes from. कभी कभी हम लोग बायस हो जाते हैं कि ये हमारी लाइन है, हम सेलेक्ट करें, लेकिन बट वी आर ऑल वर्किंग फॉर द फार्मर्स, ये ध्यान में रखना होगा. ही इज द अल्टीमेट यूजर ऑफ द टेक्नोलॉजी. एक साल आप गलत वैरायटी दे देंगे, दूसरे साल वो आपसे बात भी नहीं करेगा और फिर वो आपके साथ काम भी नहीं करेगा. तो दैट्स द की मैसेज आई वांटेड टू गिव. और यह है कि स्टिल वी हैव in our varietal composition some of the variety which are still being grown by the farmers. Few of the examples are like Swarna. When the IRRI team came and I went to one of the farmers for direct seeded rice, he was growing Swarna in 10 acres of land. And then I was surprised, we have got so many variety in the pipeline. पिछले 40-50 साल से मतलब we have not been able to replace the Swarna. चाहे स्वर्णा सबवन जीन डालें, चाहे उसमें ड्राउट क्यूटियल जाए, चाहे जो भी करें आप, स्टिल और उसकी जो इंडिविजुअल बफरिंग है, जो होम्योस्टैसिस है, वो इतनी बढ़िया है कि वो फार्मर्स आर गेटिंग एटलीस्ट मिनिमम ऑफ 4 टन. अभी हमने उनसे यील्ड नहीं चेक किया, लेकिन दैट्स बीइंग ग्रोन इन दी डायरेक्ट सीडेड राइस. सेकेंड एग्जाम्पल इज दी सम्मा मसूरी, जो पटला से ब्रीड हुई. अच्छा, पहली जो वेराइटी थी इट वाज Dr. Mr. Rao was only BSc AG. So it is not very important कि आप PhD हैं, qualified है, सब कुछ है, लेकिन but you must have an eye कि हमको क्या variety देनी है किसानों, so that you can give a better variety. Samma Masuri was a product of a student thesis. That's where I tell that the students are very important input of your ब्रीडिंग प्रोग्राम, उनको यूटिलाइज करिए. एंड देन दिस वैरायटी वाज डेवलप्ड. हालांकि एग्री प्रोग्राम से ड्रॉप हो गया था, बट समहाउ इट केम बैक. 1 मिनट. हम वो जितने स्पीकर से उतना टाइम नहीं लेंगे, बस थोड़ा सा और है, क्योंकि फिर तो पता नहीं कब दोबारा चाहे, और हम लोग 76 पार कर गए हैं, तो कब पता नहीं कब बुलावा आ जाए, फिर कहने का समय नहीं रहेगा. सम्मा मसूरी है और फिर सरजू बावन है. अभी भी हम लोग यूपी में सरजू बावन को रिप्लेस नहीं कर पाए हैं. दिस इज ए ह्यूज डिमांड ऑफ द सीड. और फॉर्चुनेटली जो सम्मा मसूरी है, इट हैज बीन नाउ रिप्लेस विद द तेलंगाना सोना, which is getting very popular thanks to the breeder who is next to our. And then we have the variety like PR126. अभी जो ब्रीडिंग मेथड की बात कर रहे थे डॉक्टर दास, दिस इज ए वैरायटी फ्रॉम चाइना डेवलप्ड बाय द हुकोंग ली, और दिस हैज इन्वॉल्व सम 8 पेरेंट्स इन द क्रॉस. अभी हम लोग बाई पेरेंटल क्रॉस कर रहे थे, 4 पेरेंट की बात कर रहे थे, एंड दे हैव इन्वॉल्व द 8 पेरेंट्स, एंड देन दे हैव सेलेक्टेड द ब्रीडिंग मेथोडोलॉजी, एंड दिस वैरायटी हैज कम, व्हिच इज नाउ गेटिंग वेरी पॉपुलर not only in the Punjab and Haryana, but Odisha also, and we have found very good in the UP also. So this is going to be a mega variety of the future. I have the enough seed, and anybody who wants, we supply the 2 kg free seed to any of the scientists. So these are some of the new things. And then in the submergent tolerant, since I have been working with the farmers, the Samba Savon has been widely accepted because of its grain quality. And the people like, because Swarna has not a better grain quality. Samba Savon is a very huge demand of the seed, and I'm not able to make it, but this is information. Anyone can multiply the seed, and I can provide the nucleus seed or the breeder seed. And then the CR-1009 Savon, because in the cyclone prone area, cyclone generally comes in the month of October, mid-October to early November. And during this period, the CR-1009 has to be at the vegetative state, so that it can tolerate the summer, the flood by the cyclone, and then it is harvested by what they call the Samba season. And then it is harvested by the February, and so that you can have a very good crop in whole of the coastal area here. So thank you very much for our listening. And thank you, Mr. Chairman, for time.
May I request
So the answer saying for his comments.
So I will not go there because if you go there, you are tempted to speak at least 5 minutes, and he told me only 2 minutes. So we'll be speaking. So, and he's right, means speaking with the giants like him, and he just mentioned that by both were the classmates in 67 and 68, and I was born in 69, so definitely I have a lot of time, though my hairs are as gray as any of them. So I will not take time, just wanted to really, first a good afternoon to all delegates, people sitting on the chairs, Pradhan Saab, and all scientists and all the guests here. That's really wonderful. And let me first congratulate Sundaram ji for first for this Diamond Jubilee celebrations of the annual RISE group meeting, organizing so wonderfully and hosting us so beautifully. So definitely entire team is deserve a big applause and congratulations from us. And we ourselves also means what in last 6 decades, whatever happened, transformative things happened in the RISE. Definitely, this annual RISE group meeting has a tremendous contribution into that. So, applause for all of those who have been the part of this group meeting. Collaboration between ERI and ICAR, really on behalf of ERI, we are very thankful for that, and this collaboration has really immensely helped in shaping number of things on rice research and deliver real impactful things on the ground also. AICRP platform is the key in taking those testing and validating number of not only the varieties but other technologies also to the different ecologies and taking them to the farmer fields also. The climate resilient varieties, healthier rice, whether is low GI or direct seeded technologies, and whatever happening as you have seen in the presentation, that all because of this eclipse testing and all the contribution of all those scientists. So definitely we are very proud of that. And by this collaboration, we are definitely aiming to contribute big. I would just highlight 2, 3 points that our leader, Dr. ML Jat, definitely has spoken yesterday. And then we are really proud that he spoke that he also belongs to the rice family. So definitely a big indication from us. We should jointly aim for 165 million tons of production by 2030 is a big target given by our leaders. So definitely a lot of onus would be on us and where these technologies should take us. Productivity increase target 3.88 or 4 ton in the short term and 6 ton by the long term is also another period that would be difficult to achieve, but definitely with those collaborations, we shall be doing all those things. Finally, I would definitely want to congratulate all those speakers from the morning. Dr. Das, the strategic vision that he showed, Dr. Mr. Pradhan, Vishwanathan ji, the Kamla story definitely is the next big things happening to our country. And I also would like to thank all those who really highlighted the importance of speed bidding in bringing all those innovations to the forefront, where that shows the CG or EG and national system collaboration can really bring such technologies that can globally lead the innovations and may target the transformative things globally. And then PVFRA and NBPGR was also very informative. So that's really sincerely thanks all of you, and I'm very much confident that together we will shape a bright future for the rice scientists as well as for the farmers also, especially the small. Thank you.
Thank you, Dr. Sir. Uh, so I see Dr. VP Singh first. His interest in rice research. ADG, your presentation was, I was listening as a student. Same thing, Dr. Das, very rich in traditional wisdom. He brought many good things, many valuable things. On the dais, I will, my colleague Dr. B.N. Singh, thank you very much. Sundaram is one very talented
leadership, very good scientist, good manager. On the dais, I see reporter Dr. Beg and reporter Dr. Infinite. I call him Dr. Infinite, as his name is Anand. And Pallavi, he has been my student. There are three students. Vikas was also, when I was director, he was here. Mal, Dr. Ashish, he was also here. And I am very happy that you people are doing excellent here. And very good presentation. And this is a special occasion. I remember in 1969, I joined AGRM program through Rockefeller Foundation with Dr. Freeman, Dr. Tanavi, Dr. Cogman. All those people were working in a partnership mode with Dr. S.V.S. Shastri and group. And it was a different working culture. From there we learned how to become field-oriented people. And mind it, any miracle will come when you will go to field, you will talk to plants, you will make the plant friends, recognize, and then product will come. It's assured that it will be commercialized. And I am grateful and remembering the contribution of all the directors over here and scientific staff I have seen how dedicated they were, and the technical staff, administrative staff. So I don't know, I have the least time, but one thing is our land is finite and fragile with multi-nutrient deficiency. Sulfur 38%, zinc 40%, iron, anything you take 6, 7. So that is the one thing. Another limitation of the climate change and adverse, So it will impact negatively, and we are expecting to feed to increase the total production. For total production, by 2030, 345, food grain, million tons. And by 250, 70% increase. And increase will come 80% by yield growth, increasing the yield. So you can see the challenge in front of you. And under this situation, you have to target the attainment of yield potential, which I think he has presented. Dr. Pradhan has already spoken about per year 1.2 million ton. And in production, I was totaling the rice and wheat, the major contribution. So things are not that easy. When you will realize the adversity and then population collision effect, probably is a big challenge to all of you. If I come to water, water has come from 1951 to 1/3. It was annual 5,777 something cubic meter, now only 1,300, 1/3. So in another 10 years, you can imagine if water is available to rise or not. Another thing, DG very he was with history, he said that we have to reduce 5 million tons, 5 million hectares. Is it true? And I think the rice family, rice community, they should think how to reduce the area. And then if you come to the other aspect of stresses, yield is limited, at see the increment, how much productivity is going on. And leave the irrigated ecosystem where you have attained 4 ton, but as a whole average is only 3 ton, much lower than the international average. So these are some of the fact things. And about the program, I have seen as chairman of this institution, wonderful work is going on. There are many products. If you go to the field, see the plant, you'll salute the workers, and they are engaged. And those who have not seen it, I'll request, please see, or sometime else you come and see it. So the institution, if you analyze impact, how much staff is there, how much budget is allocated, calculate per capita output, per scientist output, you'll find much better. I'll say almost all the ICR institutes. So I consider, if you look the impact in this, #1 institute. And I also remember the collaborative part of the IRI. We have seen Dr. Kush way of working approach. That was the time gene flow, the material was easily available. And we were working with open heart in one-to-one partnership mode. So we hope that the things should flow in the same direction. And things are changed now. We have in the market, we always find now nutritive status where there is a big lacuna. And Dr. Pradhan has presented, and that's a very valuable point. I think with these small remarks, time is lifting. So other things I'm not going to cover it. But once again, thank you, all of you, about your contribution. Thank you, doctor, you are doing excellent. And I find all my colleagues on the dais, off the dais, and salute all of you. Thank you.
Thank you so much, sir. That is truly inspiration. And we'll definitely try to follow the points which you have recommended right now from our side as an institution. Now it's time to felicitate our speakers for today's session. I request our chair and co-chair and other dignitaries to please felicitate the speakers. Our first speakers on conventional rice breeding strategies is Dr. Satyaranjan Das, Honorary Professor, OUAT, Bhubaneswar. I request sir to please come on the dais. The next speaker, Dr. S.K. Pradhan, ADG FFC ICR, New Delhi. Sir, I please request you to come on the dais, please. I request Dr. Satendra Kumar Mangrotia from ICAR-IIRR to please come onto the dais. Next we have Shri Deepal Roy Chaudhary, Joint Registrar, PPV FRA. Sir, anybody on his behalf if they can take. Or else the next is Conservation and Utilization, Dr. Vishnu Goyal, Principal Scientist, NBPGR. If sir is not there, Dr. Vikas Kumar Singh from IRI, India. Next we have Dr. R.M. Sundaram, Director, ICAR-IIRR. Okay, no problem. No, sir, this is for your speaker. Dr. Paneer Selvam from IRI, India. I request our director, sir, to please felicitate our chair, Dr. B. Mishra, sir. The session was jointly chaired by Dr. Sidhanshu Singh, IRI, India. I request, sir, to please felicitate Dr. Sidhanshu Singh, Director, IRI, India. I request Director, sir, to please felicitate Dr. B.N. Singh, sir. I thank all the delegates, chair, co-chair, conveners, and rapporteurs for this session. And we break here for lunch, but we'll come back fast. Half an hour, 30 minutes we have. And this session there is a little bit change. We'll start with awards and felicitation first, followed by the technical session. So I request everybody to please assemble within half an hour. 3:20 sharp, I request all the delegates to please assemble in the auditorium. Thank you so much. Thank you, sir.
ये बैलेंस के लिए ना हाँ वो Catch up on protein. Yeah, definitely. And you can always see that on. Yeah, super.
He's also retiring, sir. Because he'll be given a big clap now. He is not retiring. No, no, sir. My professor is retiring next March. Sir. That's why I gave such a very fondly called as Mama. And senior professor is Mama. No, name is PSPH. It was known as Mama. Mama.
and stomachful discussions. And please come and raise the dais. I also invite Dr. Aram Kumar, Chief Convener of the workshop, to please come onto the dais. I invite Dr. S.V. Sai Prasad and Dr. C.H. Padmavati, ICAR-IIRR, to act as rapporteurs for this session. We have a small change in our program and we would like to have a very interesting session right now. So we are going to start with some of the awards during this Diamond Jubilee celebrations. We would like to honor some of our greats and we would start with the Lifetime Achievement Awards. Sir, would you like to? Okay. Dr. Kalyani will now take over the session. Sir, we would like to felicitate Dr. B. Mishra, sir, former VC, SKUAST, Jammu and Kashmir. Sir was our former director of BWR as well as DRR, Directorate of Rice Research, and he has been a pioneer in developing rice varieties specifically for salinity tolerance in rice. And he is a visionary. Today also we had an opportunity to listen to him. So we thank you, sir. He is coming. Next, we would like to call upon Dr. B.N. Singh, sir, former director, ICAR-IIRR Cuttack. Dr. B.N. Singh, sir. Dr. B.N. Singh, sir, has been pivotal in developing rice varieties, specifically for submergence tolerance. And we have been known him for taking the varieties to the farmers through extensive extension. Sir, please, sir. We would now like to call upon Padma Shri Dr. Vijaypal Singh, sir, who was former program leader. We would like to call upon Padma Shri Dr. Vijaypal Singh, former program leader Rice, ICAR-IIRR New Delhi. He is well known for his variety Pusa Basmati 1121, which has fetched India a lot of export amounts and money. So we would like to honor you, sir, here. And for his great work in Basmati, he was also honored by the Government of India in 2012 by the highest Indian civilian award of Padma Shri. GA Parade. Thank you so much, sir. Kaushik, please. We would like to now call upon Dr. G.A. Parade, Ghulam Ahmad Parade from Emeritus Scientist, SKUAST, Jammu and Kashmir. Sir, he is a principal breeder for Shalimar Rice 2 variety. Sir, so he is an associate director of research at Rice Research and Registration Station in Khudwani, Anantnag, Jammu and Kashmir, and he has been known for his contribution as a plant breeder and genetics in Jammu and Kashmir specifically. Thank you so much, sir, for your contributions in rice. Now we would like to call upon to our own Dr. A.K. Singh, sir. He's an Indian scientist and plant breeder, plant geneticist, a great teacher, and his work in basmati rice has been pioneering. Sir has also received several awards for his services to the society, and he's a main person who was like behind for marker assisted plant breeding in India. Now we would like to felicitate again our own Dr. V. Ravindra Babu sir, who was former director ICAR-IIRR Hyderabad, and the institute name change was also like it happened during his regime, and he is very well known for rice high zinc varieties. So we thank you, sir, and take this opportunity to extend our gratitude. Thank you so much, sir. Now we would like to call upon for our own honorary professor whom we had heard little time back, Dr. S.R. Das, OUAT Bhubaneswar. So sir is associated with studies as well as releasing many breeding lines, rice breeding lines, which is tolerant to submergence and stagnant flooding. He is associated with 49 high yielding rice varieties in rice. He is also a recipient of Sinadhira Rice Research Award 2012 conferred by IRI. Thank you so much, sir. Thank you, sir. Now we would like to call upon Dr. Deepak Sharma, who is working as Head Department of Genetics and Plant Breeding, IGKV Raipur. Sir has developed rice hybrids as well as the mutant rice lines specifically for land races of Chhattisgarh. And he is also working as a nodal officer for PPV FRA for conservation and utilization of crop biodiversity. Also, he has facilitated in securing geographical indicators, that is the GI tags, for two popular traditional rice varieties, Jeera Phool and Nagraj Dubra. So, thank you so much, sir, for your contribution to rice. Now we would like to call upon Dr. S. Thirumeni, Professor of Plant Breeding and Genetics, Pajankova, Pandit Jawaharlal Nehru College of Agriculture and Research, Karaikal, Puducherry. Sir is a professor in plant breeding and genetics, and he is going to retire this year. He has been instrumental in developing many submergence tolerant rice varieties, specifically suitable for Karaikal and southern India. His, his 3 to 4 varieties were released last year and dedicated to the nation by Honorable Prime Minister of India. Yeah. Now comes the most awaited center award. Uh, I would like to say that all of you stand equal, maybe a little more. We are trying to award some of them who have put in some extra effort. So please do not think that if you are not awarded, that you do not deserve an award. So the first best overall AGRIP center funded goes to the main rice research center, Navsari Agriculture University. We also have another best overall AGRIP center funded, which goes to the Rice Research Station, Raipur, Indira Gandhi Krishi Vishwavidyalaya, Chhattisgarh. We would also like to appreciate the efforts of a voluntary center. And so the best overall AGRIP center voluntary goes to Pandit Jawaharlal Nehru College of Agriculture and Research Institute, Pajankova, and RI Karaikal, Puducherry. Let's put our hands together for the awardees. Now we move on to crop improvement. The best AGRIP center in crop improvement funded center goes to the main rice research station, Navsari Agriculture University, Gujarat. The best AGRIP center for crop improvement funded goes to the main rice research center, Navsari Agriculture University, Gujarat.
Again, the best equipped center for crop improvement. The second institute is the rice, not second, it shares equal, the rice research station Raipur, Indira Gandhi Krishi Vishwavidyalaya, Chhattisgarh. I would like to say that the individual uh, division awards have been assessed separately and then the combination has been given. Each division has constituted their own committee and then they have assessed the applications. And now following a similar trend, we have the best equipped center for crop improvement in the voluntary category going to Pandit Jawaharlal Nehru College of Agriculture and Research Institute, Pajankova and RI, Karaikal, Puducherry. The next group of awards is for agronomy. And the best equipped center for agronomy goes to Govind Ballabh Pant University of Agriculture and Technology, Pantnagar, Uttarakhand. Again, this award is being shared by another center, and the best equipped center agronomy goes to Tamil Nadu Rice Research Institute, TNRRI, Adudurai, Tamil Nadu Agriculture University. Adudurai Center. We have only one representative from Madurai. Everybody from the center can please join. And the best equipped center agronomy in the voluntary category again goes to Pandit Jawaharlal Nehru College of Agriculture and Research Institute, Karaikal, Puducherry. please remain on stage. Please, all of you remain on stage. The best equip center soil science in the funded category again goes to Pandit Jawaharlal Nehru College of Agriculture and Research Institute, Karaikal, Puducherry. And this award is being shared by another center, Regional Agricultural Research Station, Maruteru, Acharya N.G. Ranga Agriculture University, Andhra Pradesh. The best equipped center for soil science. Everybody from the center, please join. The best equipped center soil science in the voluntary category goes to the Mountain Research Center for Field Crops, Kudwani, Shari Kashmir University of Agriculture, Science and Technology, Kashmir. We move on to physiology now, and the best equipped center in the funded category for physiology goes to the Regional Agricultural Research Station, Maruteru, Acharya N.G. Ranga Agriculture University, Andhra Pradesh. Please put your hands together for the awardees. Oh, this is a wonderful group. I'm loving to see this. And this award is also shared by the Regional Agricultural Station, Kharjat, Dr. Bala Saheb Sawant, Konkan Krishi Vidyapith, Maharashtra. The best equipped center physiology in the funded category goes to the Regional Agricultural Research Station, Kharjat. The best equipped center physiology in the voluntary category again goes to Pandit Jawaharlal Nehru College of Agriculture and Research Institute, Karaikal, Puducherry. We now move on to the next subject, pathology, and the best ACRIP center in the funded category goes to the Agricultural Research Station, Gangavati, University of Agricultural Sciences, Karnataka. Again, this is shared by another center, the Main Rice Research Center, Navsari Agriculture University, Gujarat. The best ACRIP center, pathology. The best ACRIP center, pathology in the voluntary category goes to the Hybrid Rice Evaluation Center, Gudalur, Tamil Nadu Agriculture University. The best ACRIP center, pathology in the voluntary category goes to the Hybrid Rice Evaluation Center, Gudalur. Please put your hands together for the awardees. We now come to the next subject, entomology, and the best ACRIP center in the funded category goes to Regional Agriculture Research Station, Pattambi, Kerala Agriculture University. And this award is again shared by another center, the Rice Research Institute, Rajendra Nagar, Professor Jayashankar, Telangana Agriculture University. The Rice Research Institute, Rajendra Nagar. Please put your hands together for the awardees. We also have in the voluntary category, again, the Pandit Jawaharlal Nehru College of Agriculture Research Institute, Karaikal, Puducherry. The best ACRIP center in entomology again goes to Karaikal, Puducherry. The best ACRIP center in the voluntary category for entomology goes to Pandit Jawaharlal Nehru College of Agriculture and Research Institute, Sajankova and RI, Karaikal, Puducherry.
We also have special awards for different ecologies. And the best center for the significant contributions made in coastal rice ecology goes to Agricultural Research Station Ragolu, Acharya N.G. Ranga Agriculture University, Andhra Pradesh. The best center for significant contributions made in coastal rice ecology goes to the Agricultural Research Station Ragolu, Acharya N.G. Ranga Agriculture University, Andhra Pradesh. And the best center for the significant contributions made in hill rice ecology goes to Mountain Research Center for Field Crops, Kudwani, Sher-e Kashmir University of Agriculture Sciences and Technology, Kashmir, Jammu and Kashmir. Congratulations to all the awardees. Being a part of this RISE network itself is an award, I think, and so nobody is left behind. Now this is a time to felicitate our retiring scientists, retiring ICRIP scientists from various centers. So to begin with, Dr. M.J. Baig sir. Dr. M.J. Baig sir is currently working as a director in charge at Central Rice Research Institute, Qatar. He has a research findings and focused on the physiological analysis of grain yield in hybrid rice, screening crops for drought and salinity tolerance, and studying the effect of elevated CO2s and share on crop physiology and productivity. He has more than 80 research papers, 16 book chapters. He has received several awards like ICAR Senior Fellow Researcher and top many merit lists. Thank you so much, sir. Thank you for all your contributions. Now we would like to felicitate Dr. Sangamitra Samantre. Madam. We'll just read out. Dr. Sangamitra Samantre was from Qatar and she has joined by ICR in 2010. Her research focuses on double haploid techniques. Madam has significantly contributed to hybrid rice breeding, leading to the development of 3 high-yielding rice hybrids, CR1, 702, 703, 704, and she is very well known for double haploid anther culture as well in rice. So we thank the contributions made by Madam Sangamitra Samantre. Now we would like to call upon Dr. S. Tirumani, Professor of Plant Breeding and Genetics from Pandit Jawaharlal Nehru College of Agriculture and Research Institute, Karaikal. So, sir is basically a teacher and he has taught genetics and plant breeding to all of his students. Plus, he is also a researcher and he has been instrumental in development of several promising salt and submergence tolerant rice strains and release of 6 crop varieties in Kumbu Napier hybrid grass and 5 in rice. Also, he is instrumental in releasing and developing marker assisted selection lines for rice. Thank you, sir. Narasimha. Now we would like to felicitate Dr. R. Narasimhan. Narasimhan is from KVK Puducherry. Dr. R. Narasimhan completed his studies from TNIU and he has been appointed as Junior Rice Breeder at the Peruntalaivar Kamaraj Krishi Vigyan Kendra, Puducherry. His field of specialization includes application of in vitro culture techniques in plant breeding with special focus on development and evaluation of endoclonal variants of Ponni rice cultivar. I would also like to add that he is my good friend from 1983. He is my classmate and my... Please, ma'am, please. We also request Dr. Vinod to please join on the stage. Thank you, sir. Thank you so much. Now we would like to felicitate Dr. Subrata Ray, ex-Assistant Director, Department of Agriculture and Farmers Welfare, Government of Tripura. Dr. Subrata Ray. Okay, sir. Okay, sir. Then we would like to call upon Dr. Dhirendra Singh. Okay. Dr. Subrata Ray has been working in the College of Agriculture, Tripura as a senior scientist plant breeding under the coordinated project AICRPR on various crops. Thank you, sir. Now we would like to call upon Dr. Dhirendra Singh, Associate Director cum Principal Scientist, HAREC, Dhaulapua. Sir is there. Then our own Dr. R. Mahendra Kumar. So sir is Principal Scientist and Head Agronomy. He is PI Agronomy as well from our institute and AICRPR PI also. Sir's basic interest in rice. All heads of ICAR-IIRR, we request you to please join on the dais. Sir is associated with the development of nutrient use efficiency in rice as well as pest disease resistance. He has also developed SRI techniques and taken many demonstrations of direct seeded rice. He got advanced training in biochar utilization in rice as well. And recently, sir has got a project from SBI Foundation under CSIR grant of 4.5 crores for DSR promotion. So we congratulate R. Mahendra Kumar sir. Next we call upon Sureka Madam on the stage. She has a significant contributions in the agri for the past 32 years. She is known for her research in the area of nutrient and soil management aspects, SSMM of rice, hybrid rice, SRI, crop residue management, organic farming and nutrient use efficiency. She has taken various trials under nutrient use efficiency, especially nitrogen, evaluated popular genotypes and improved lines under NUE for both soil and applied nitrogen utilization. Now we call upon Dr. P. Raghuveera sir. Dr. Pushkar Raghuveera sir has an expertise on seed physiology, accelerated aging, impact of climate change on croplands, direct seeded rice, new plant types and idiotypes on rice crop physiology, source sink relation systems, and system of rice intensification and mineral nutrition. It has a great contributions in the role of silicon in inducing the biotic and abiotic stresses tolerance in rice. Next, we would like to call upon our dear Dr. P. Muthuraman sir. Palaiva. He is the heart and soul of IIRR. He is the one who knits everybody together. In addition to that, he is having 8 research projects. He has organized more than 200 rights-related subject matter training programs and trained nearly 9,000 extension functionaries and scientists. Your young person, sir, will be very sorely missed in the institute. Next, we call upon Dr. N. Nagarajan from Auditorium. Dr. N. Nagarajan from Auditorium. Anyone from the Auditorium can join. Yes. Has done extensive work in instrumentalization of development of 22 rice varieties, including many basmati rice varieties, which have collaborated for the 48,000 crore foreign exchange earnings. And also he had 120 research projects, and he was involved in the several thermosensitive genetic male sterile lines. As a lead breeder, he has developed 2 TGMS-based rice hybrids, Pusa JRH56 and Pusa RH60, making the first of its kind in the public sector hybrid breeding in the country. Now we are coming back to the scientific mode. We will have presentations of the VIC recommendations. I now invite Dr. Sai Prasad to please present the VIC recommendations along with Dr. Hari Prasad.
Good afternoon, everybody. Sir, I will give a summary of VIC meeting of which had been occurred on 26th April. We have received around total number of varieties around 35 under irrigated under varietal this thing, in which 26 were identified. There are 7 varieties have been identified under ETP early transplanted, that are IET 30642, 30641, 30669, 30636, 29940, 30656, 32036 under this category. And under category irrigated medium early, there we have it was identified 3 varieties have been identified, in which 30756 and 32045, and one variety 32043, which is a genome edited. This is the first time it was released and the generated in the background of MTU 1010 from IRI New Delhi. In the next category, irrigated medium, only one variety was identified, that is 32051. And under MS, there are four varieties have been identified, in which 30961, 32054, 32056 have been identified, and another variety 32072, which is awaited since long term, that is a denominated with the background of BPT 5204, which is a high yielding around more than 17 to 18% higher yield than the BPT 5204, which is denominated product. And under LPT, low phosphorus tolerance, there are two varieties have been identified, that is 31110, 31110 and 28070. These two varieties have been identified under LPT. And under LNT, low nitrogen tolerance, it is three varieties have been identified, that means 29581, 28084, 29578. These three varieties have been identified under low nitrogen tolerance. And one variety has been in saline and alkaline ALS ASTVT, 31055 was identified. And there are two varieties under basmati, that is identified 30533 and 31307. And under late conditions, irrigated late conditions, only two varieties have been identified, 32064 and 32123. Under EDS, early drilling system, the 26, one variety was identified, that is 30336. These are the 26 varieties have been identified, and four varieties have been deferred because of some quality traits and other components, that are IET 30772, IET 30819, 32047 and 32061. And five varieties have been non-identified. And one variety IET 29975 has been recommended for area extension. They can again submitted for area extension because it has already been released by SVRC. This is about the gist of this, and it is not at final. We will take the permission of the DG and we will put it on online, the VIC report. Thank you very much.
Very shortly, it will be put online with complete details. It has gone to competent authority for approval. Okay.
Respected Chair, Honorable Director, and dear delegates, very good evening. In hybrid rice, 21 proposals were submitted, and all 21 proposals were examined by the VIC committee. In this irrigated early ecology, 5 hybrid entries they were recommended. They are IET number 30561. IET 28956, IET 30565, IET 29694, IET 29700. In mid-early, total 7 applications were examined. In these 4 were recommended. They are IET 30593, 30579, 30574, 30573. In irrigated medium, 5 proposals were examined, all 5 were recommended. They are IET numbers 30613, 30603, 30605, 30604 and 30608. In early direct seeding ecology, one proposal was submitted and it was recommended. The IET number is 31193. And in saline and alkaline ecology, one proposal was submitted and it was recommended. Its IET number is 30165. The 4 proposals were not identified. They are IET number 30555, 30575, 30578, 30589. One proposal IET 28579 deferred. Among all the recommended proposals, 2 are from public sector institutions, one from Central Rice Research Institute, Cuttack, one from my institute, Indian Institute of Rice Research. I congratulate all the bidders who have submitted their applications for this VAC. Thank you.
I now invite Dr.
Uh, Dear participants, these are the provisionally identified, yet to be approved by the council. Detail they will be sending, and we will be verifying all the things. Then soon we are going to give a clear picture, and director IIRR will be uploading in their site. So these are only provisionally clear. Congratulations to all the lead developer, co-developer and associate. Congratulations.
I now invite Dr. S. V. Sai Prasad again to present the recommendations of plan reading section.
Sir, we are discussing since last honorable chairman and co-chairs and other things. I want to, because 3 days we are discussing about the various activities of plan bidding, and these are some of the recommendations which are given under the leadership of Chairman Dr. A.K. Singh. When an entry is performing on overall basis but has significant advantage only in few zones, but not in all zones, the entry will be promoted only in those zones where it has significant yield advantage. Second one is in addition to the grain quality analysis at ICAR-IIRR, satellite labs at 3 centers Raipur IGKB, Navsari NAU and Rajan Nagar PGTAU were identified, which will cooperate in grain and nutrition quality of a set of equipped trials. Soil samples should be analyzed to assess the nutrient status at location conducting LPT and LNT trials. Accordingly, the promotion of entries should be done. Any center volunteering to conduct trials should also take up complete set of IVT, AVT1 and AVT2 of an ecology. As far as funded centers are concerned, the trials should be allotted based on mandate suitability of the region. For promotion of hybrid or variety, entries in AICR-IIRR, ECRIP project on rice, they should meet the requirement of more than 67% milling percentage and also 55% XRR. Breeding of herbicide tolerant rice should be restricted to early direct seeded rice, DSR ecosystems only. Furthermore, the target area should be limited to regions where the original parent variety is already recommended for cultivation. Thank you very much.
I now invite Dr. R. Mahendra Kumar to present the recommendations of agronomy.
On the lighter side, I don't know whether after taking retirement function, whether I present or not. Anyhow, very good evening to everyone. I will briefly, I think, what are the decisions taken our group meetings 26 and 27. This is, I think, already highlighted in the morning also. All AVT2 entries has to be sent to agronomy by May 5th. May 15th. So beyond that, we are not going to conduct that AVT2 trial for those entries. Maybe kindly noted down. We have some recommendations by our chair and co-chair. They are very emphasizing on quantification of the water for the different crop establishment method, especially for direct seeded rice. And now we are planning to go for IoT based water management, which we can online recording will be done. It was also suggested to have a long-term trials on weed shift in the wet and dry DSR system. And there was a problem of, I think, VD rice in future. So there will be trial on VD rice, which is, I think, proposed by Kerala Agriculture University. We wanted to increase separate the yield maximization in the direct seeded condition because there are a lot of reductions in the direct seeded rice. And there will be maximization trial for direct seeded rice. And our chair also suggested to include biostimulants and some climate resilient products like clean rice for the reducing the climate effects of the rice cultivation. Based on our trials conducted during 2024, we found that the trial which was conducted for more than 3 years, the recommendations are alternate wetting and drying was proved very efficient in terms of climate resilience. Also, reducing 30 to 40% of the water for the rice cultivation. We could able to see that still we need to apply 125% of recommended dose of fertilizer in addition to the farm manure for enhancing the higher yields in case of irrigated rice system. As for last 3-4 seasons, IPM is I think very effective and it has improved yields from 10 to more than 23% wherever IPM is practiced, especially for the weed management. And fourth recommendation is we could able to see that puddle and unpuddled systems are on par in terms of grain yield in some of the zones, and we can effectively manage weeds with mechanical weeder. These are the things, sir, recommendations of agronomy. Thank you, sir. Thank you. I would like to thank my cooperator. This may be, I think, last under equip program. I would like to thank everyone.
I now invite Dr. MBB Prasad Babu to present the recommendations of soil science.
Respected Chair, co-chairs, the soil science group meeting recommended residue application at 5 tons per hectare can help in saving of 25% nitrogen and 100% total 100% of care requirement in addition to enhancing soil organic carbon content. And the second one is long-term application of either FIM 10 tons per hectare alone or 5 tons per hectare in addition to RDF can significantly improve soil fertility parameters like soil organic carbon, available nitrogen, phosphorus and potassium status. Third one is integrated crop management with need-based pest control can result in a higher grain yield of up to 35% for complete natural farming. The group meeting, the group discussions have recommended the following action points, 2 action points. The soil microbiome to be assessed through metagenomics in long-term fertilizer trial, and then the impact of nano-urea application on long-term productivity and soil nitrogen status is to be explored, studied. Thank you.
So all your proceeding is approved. That's it.
One more, 2 more.
I now invite Dr. P. Raghuveer Rao Garu to come and present the plant physiology recommendation.
Respected Chairman, sir, and co-chairman, and Dr. Baig, sir, and all my colleagues on the dais and off the dais. Now I stand before you to present the recommendations and salient findings of ICAR plant physiology and biochemistry for the Kharif 2024. Sir, our major effort was to find out which are the donors for major abiotic stress tolerance, along with a mechanism which can help with breeders in identifying suitable donors for abiotic stress tolerance. The first one is the role of silicon in inducing abiotic stress tolerance, sir. We found that it ameliorates moisture stress and can be used as a very good stimulant for producing enzymes. Second one is phenotyping a varied rice genotype for drought tolerance. The mean grain yield of rice reduced by 20 to 23%, and we identified D1633 followed by RP6469 as donors for rain-fed upland situation. Screening for high temperature stress tolerance, sir. There was a reduction of 30% yield reduction because of high temperature imposed by the experiments, and we found that IET 31512 and NDR 97 as useful donors. As physiological calculation of selected genotypes for multiple abiotic stress tolerance, we found that very good genotypes have been identified for multiple abiotic stress tolerance, particularly for salinity, osmotic stress, and also anaerobic germination. Raspanjor and Ratnagiri 8 are such varieties for multiple abiotic stress tolerance. Screening for rice germplasm for submergence tolerance, we found that CBM AS 22061 is highly tolerant as compared to FR13A. And coming to low light stress tolerance, sir, along with Swarnaprabha, we also identified IET 31246 and IET 32147 as good tolerant checks for Swarnaprabha and low light stress tolerance. Thank you, sir.
Dr. Baig is approved. Approved.
Can we go to the next one, sir? So I would now like to invite Dr. A.P. Padma Kumari to present the recommendations of entomology. Mr. Chair, dignitaries on the dais of the dais and fellow scientific fraternity. Based on the deliberations in the entomology group meeting, it was decided from the specific trials in the plant hopper screening trial, 8 genotypes were identified as promising, that is MTU 2721-7-1-21, MTU 272028-2-1-1, MTU 2721-7-1-2-2, RP6740 SPM MS70, MTU 2716 28-2-1-2, MTU 2716 20-2-2-2, RP5977-MS 112. Then ISM B8 and JGS B8935 as promising. They will be promoted to the MRSC screening trial and they can be registered as donors for plant hoppers. In the second year of Garnet screening trial, 4 genotypes were identified, that is ISM 24, WGL 1909, RGL 294, and RMS ISM 26, and they can be promoted to the multiple resistance screen trial and can be registered as donors for Garnet resistance. In the second year of testing for Stemboro screening trial, 4 entries, that is RP4919 NSR24, BK49-76, RP650540, RP6112 SM92R293 were found promising for Stemboro tolerance. They will be promoted to the MRST trial and can be registered as donors for Stemboro tolerance. And NND2 Elanders was found promising in the MRST trial and it can be suggested for registration. Thank you.
Entomology group, I hope we should approve. Approved.
Now invite Dr. M. Srinivas Prasad to present the plant pathology recommendations.
Respected chairman, co-chairman, and then respected dignitaries on the dais and off the dais. The pathology group deliberated and then brought some few recommendations. We have identified several genotypes that are having multiple disease resistant lines, like CBMA-SP and the GLB94, ISHP30, and then JGL 47870, and then N4933. These were resistant more than 4 lines. That's why several lines are there, but we have given these recommendations where they are showing more than 4 lines. It is written in the recommendation. Now, another in the management aspects, a new combi product like Azathiostrobin, Tebuconazole, and Procrolizole at the rate of 3.5 was showing broad spectrum of managing the disease against all fungal diseases. That's why we are recommending from that group. Thank you.
Pathology group, I hope we should approve. Yes, approved. But one thing, these are not included in the draft. Draft write-up, you have to complete it. Okay. Finally,
Thank you, sir. Thank you, madam. Sir, now we would like to read upon the rapporteurs list and we have a certificate for them. So quickly we can give the certificates. So first I would like to call upon Dr. Lada Lakshmi, ICAR-IIRR. I request all the rapporteurs to please be ready and quickly so that we can save some time. Dr. G.S. Jesudasu, ICAR-IIRR. Dr. M.S. Ananta, ICAR-IIRR. Dr. Divya Balakrishnan, ICAR-IIRR. Dr. Gobinath, ICAR-IIRR. Dr. Mangaldeep Tuti, ICAR-IIRR. Dr. K. Sunita, ICAR-IIRR. Dr. R. Abdul Fias. Okay. Dr. Abdul Fias. Dr. Spandana Bhatt, PGTAU ARI. Dr. Spandana Bhatt, Dr. V. Mansa, ICAR-IIRR. We thank all the rapporteurs for promptly recording all the proceedings and submitting it to us within time. Dr. Arvind Kumar, ICAR-IIRR. Dr. R.L. Verma, CRRI Cuttack. Dr. R.L. Verma. I request anybody from CRRI Cuttack to please collect the certificate. Dr. R.L. Verma, CRRI Cuttack. Dr. Harita, IRI, New Delhi. Dr. Harita B, IRI, New Delhi. Dr. Harita. Yes, sir. I request Dr. Kaushik Chakrabarti from CRRI Cuttack to please come on the dais. If he is not there, anybody from sir, CRRI Cuttack for Dr. Kaushik Chakrabarti. Dr. Guru Prasanna Pandit, CRRI Cuttack. Dr. S.V. Sai Prasad, ICAR-IIRR. Dr. S.V. Sai Prasad sir from ICAR-IIRR. Dr. P.K. Bhaumik from IRI, New Delhi. Dr. P.K. Bhaumik. Anybody from team IRI can collect the certificate. Dr. Nikhil Kumar Singh, BHU. Dr. Nikhil Kumar Singh, BHU. Dr. Lambodar Behra, CRRI Cuttack. Dr. Chinnababu Naik, ICAR-IIRR. Dr. Sanju Thorat, Anand Agricultural University, Navsari. Navsari Center. Sanjay. P.K. Bhaumik, Dr. P.K. Bhaumik, IRI, New Delhi. Dr. Sanju S. Thorat, Main Rice Research Center, Navsari, AAU1. Dr. Baswaraj, ICAR-IIRR. Dr. Baswaraj. Dr. Baswaraj. Dr. Jyoti Badri, ICAR-IIRR. Dr. Bandappa, ICAR-IIRR. Dr. Suvarna Rani, ICAR-IIRR. Dr. C.H. Padmavati, ICAR-IIRR. Suvarna. Dr. Akshay Sakre, ICAR-IIRR. Dr. C.H. Srinivas, Angrove, Maruteru. Anybody from team Maruteru can please come and collect the. Dr. C.A. Srinivas, Angrove, Maruteru. Dr. S.L. Krishnamurthy, ICAR-IIRR. Dr. V. Prakasham, ICAR-IIRR. We would also like to felicitate all. So conveners of this 68th ARGM are Dr. R. Mahendra Kumar. Co-conveners are Dr. M. Srinivas Prasad, Dr. S.V. Sai Prasad, Dr. M.B.B. Prasad Babu. So here we have all the committees which were made specifically for this ARGM. So I would like to call upon all the committee chairperson and co-chair onto the dais. We can have a group photo. I'll just read the names. Accommodation Committee, Chairman Dr. Brajendra, Co-Chairman Dr. S. Arun Kumar. Registration Committee, Chairman Dr. D. Krishnaveni, Co-Chairman Dr. Shitanshu Kumar. Can we have them on the dais, please? Audio Visual Committee, Chairman Dr. B. Shailaja, Co-Chairperson Dr. Jay Kumar. Program Hall Arrangement Committee, Dr. Amtul Waris, Co-Chairperson Dr. Chitra Shankar. Finance Committee Chairman Dr. A. Hariprasad, Co-Chairperson Dr. M.S. Ananta. Proceedings Committee Chairman Dr. C.H. Padmavati, Co-Chairperson Dr. S.V. Sai Prasad, Dr. A.S. Hariprasad. Transport Committee Chairman Dr. P. Raghuveer Rao, Co-Chairperson Shri Shitanshu Kumar. Hospitality and Food Committee Chairman Dr. Y. Sridhar, Co-Chairperson Dr. Bandappa. Press Publicity and Exhibition Committee Chairman Dr. P. Muthuraman, Co-Chairperson Amtul Varis. Publication Committee Chairman Dr. K. Surekha, Co-Chairman Dr. C. N. Neeraja. Medical Facilities Committee Chairman Shri Shitanshu Kumar, Co-Chairperson Dr. Mangaldeep Tupi. So we thank each and every one of these committees that these 3 days went on so smoothly and like very easily because of this chairpersons and co-chairperson and the entire team who are behind the 60th ARGM. So I request everyone to please give a big round of applause for everyone on the dais. We can have a group photo here. We also thank the organizing committee, also thanks all the team members. Now we have the address by our chairperson, Dr. S.K. Pradhan sir. So I request sir. आपका address.
Good evening, all. First of all, I congratulate all those breeders, lead breeders, and the whole team for release of their varieties. Congratulations to all. Also, I congratulate to the winners, award winners, particularly Best Center, then others, winning teams for ECRIP awards. Congratulations to all the team. So finally we come to the closing session wherein last 3 days we thoroughly actually discussed particularly about review of our result, last year result. Also, we planned our experiment for next year. So I feel that this is a very successful one where I saw the participation of all the delegates or any change of the plan, yeah, any decision we took here, that was a total participatory type, and we got input from all the participants for all the decision we took here. So I profusely thank you all for taking active participation in the workshop and making it a very successful one. Actually, this is really, I feel, a great feeling of what Dr. V.N. Singh was telling. It's a reception by IIRR, and same thing I am feeling last 3 days. Actually, this is a reception to our whole ACRIP team for the Diamond Jubilee celebration. It is like a reception party to all of the center. Congratulations to the leader and all the organizing team. So it is a great success, great event. We also hope the same in future also. So thank you all. I thank the various chairmen and committee members who I was seeing very actively they were participating and they were doing all the function of the program so efficiently, we could not know any lapses from our side. So a great congratulations to the team, whole team of IRR and the participating team. At the end, I don't want to prolong more. At the end, I want to say few of the home taking message, what we should be ready for the future. So that is, we can say few point wise, I can say few points here so that you will be, while returning back, you will be just taking with you few suggestions what we did during last 3 days. First of all, you have to you have to think our target the way we discussed, that is our target to be a Viksit Bharat, and this is the main food crop. You know, this is the food security crop. So the challenges we discussed last 3 days and we planned the activity. So accordingly you takes very appropriate step, particularly you know all the technology and varieties we are depending on your result. So please be sure that whatever material is nominated, you please take it very carefully. Main point is that it's right environment first we should put, right target environment. If a drought material you are putting under submergence will be futile exercise. So please take care, you know, so much of effort leader is giving, unless you evaluate properly in a proper environment, everything will be lost. So to encourage our breeders, motivate them, your support and help is highly required. So I hope you will do this secret work, a priority work. You should not do at the end. Wherever time is available with you, you should not conduct the agreed trial. It is the priority, right time, right time you should conduct the trial. Second input I want to give you, that time has gone. Professor Das was also telling, so many varieties, multiple crossing, there is need of now. So you think of doing multi-trait breeding. Already we discussed a lot about that. We don't need one trait in one promising genotype. We need many together and simultaneously in a promising background, superior one. So please think that how to do it. If not today, you have to do it tomorrow. So already I congratulate and thanks IIRR and NRRI. They have already started multi-trait breeding, particularly what I was referring to genomic selection. So you all, wherever there is a very practical approach, I told, wherever less resources to be utilized, I showed the flow diagram. So accordingly, you please think that. So next is let us hope for the next year best result from you all, and like this year all the centers they performed well. I have reviewed 2 days 15 and 6, I think 16 and 17 individual centers I reviewed, and actually very satisfactory. Few centers they are outstandingly they are doing well. So let us congratulate the outstanding centers. And uh, I also say there are many centers who are doing good. So also we congratulate them to be outstanding. And there are few centers where the progress is not so good. We request them to be at par, yeah, at least see the others' performance and improve your center's performance. So that is in nutshell. These are my 3 4 home taking message. Please review yourself and expect best for the next year. See you for the next year program and review, and wish a very happy journey and safe return. Thank you all.
So we have a small request, sir. Dr. Deepak Sharma ji would like to hand over his publications to you personally. Sir, Dr. Pradhan ji. This brings us to the close of our 3-day workshop on Diamond Jubilee 60th.
Before we go for the last item, vote of thanks, there is one thing, because Dr. Beg, my colleague from NRRI, he has served the AGRM and also the institute for many long years. Sir, we would like to have a small address from your side.
Thank you very much, Dr. Sundaram. Today's chairman, Dr. S.K. Pradhan, ADG FFC, and Dr. Sundaram, director, Dr. Mahendra Kumar, and my colleague Sai Prasad and Padmavati, and other participants, Dr. Mitra. So many participants have left this hall, and very thin attendance. At the outset, I congratulate all the award winners and centers who have been awarded for their excellent achievements in the last year. So I congratulate all those participants. and I congratulate all those individuals who have been awarded and felicitated. I am also in that category. I have also been felicitated for my superannuation in the next month. I am going to superannuate in the next month, so I am extend my sincere thanks to organizer for felicitating me in this platform. And as we are at the concluding session of this Diamond Jubilee annual Rice Group meeting, I stand here with immense gratitude. The past 3 days have been a testament to the extraordinary commitment of the AGRM Rice Network, ICAR institutions, and our global partnership in manufacturing rice research for national and global food security. Why is that global? Because India is now the largest exporter of rice and contributes to the food security to many nations. Indian earned 10.41 billion US dollar in 2023-24. I commend the rapporteurs, session chairs, and conveners for steering insightful deliberations and wonderfully managing the whole program. Special recognition to the AGRM centers receiving awards. Today, your dedication and commendable. Let us also honor our retiring scientists who have also honored in this platform, whose dedication services have laid the fundamentals for the milestone of celebration. The recommendations presented today must guide our roadmap for 2025-26. As ICAR-IIRR, we reform our commitment to collaborative research, bringing gaps between laboratories and farms, fostering public-private partnership to scale these innovations, along with several outstanding varieties developed through conventional methods identified together first time in the history. We have identified 2 genome-edited varieties. I congratulate the developers and the team members who have developed these genome-edited varieties. As we align with the vision Atmanirbhar Bharat and the sustainable development goals, let this Diamond Jubilee be a catalyst for action, climate change, resource scarcity, and nutritional security are not challenging. We can see them as opportunities to innovate, the adapt. In closing, I extend my deepest appreciation to ICAR-IIRR for holding this Diamond Jubilee. Let us return to our institutions with renewed energy, fortified by shared knowledge and unified purposes. And I again congratulate the whole team of IRR for holding this mega event, 60th Rice Group meeting and Diamond Jubilee celebration. I extend my sincere congratulations to all the members of this IRR. Thank you very much. Jai Hind.
Thank you so much, sir, for your address and kind remarks. I now invite Dr. M.B.B. Prasad Babu to propose a formal vote of thanks.
Um, Continuing forward by our excellent wishes and comments by Director NRRI, I'd just like to summarize past 3 days events as outstanding, and cooperation from each one of you was also outstanding. And let's stand committed to work for the benefit of our country's rice farmers and also consumers, not only in India, across the world. And thank you one and all, and thank you very much. And thanks are also due to our Honorable Director General, Dr. M.L. Jack, Honorable Padma Vibhushan, Dr. R.S. Paroda ji, and also Dr. D.K. Adava ji, Deputy Director General, and most importantly, our beloved ADG, Dr. FFC, Dr. S.K. Pradhan. Their cooperation in terms of fine-tuning the program and then organizing this event in the best possible way has been outstanding. I thank all our guiding fathers who stood along with us in terms of guiding us as students, but also as mentors. I thank each one of you for being with us, and let's all stand committed together to take our right science forward. Thank you very much.
Sir, I can go from here. I will not be using. Honorable Chair Dr. S.K. Pradhan, ADGFFC, esteemed co-chairs Dr. R.M. Sundaram, Director IRR, and Dr. M.J. Baig, Director CRRI, distinguished delegates, my dedicated organizing committee members, and all those present here today. On behalf of the entire fraternity of the All India Coordinated Research Project on Rice, and on my personal behalf, it is my privilege to extend a heartfelt vote of thanks to everyone who contributed to making this event a remarkable success. We begin by expressing our deepest gratitude to the respected chair, Dr. S.K. Pradhan, whose leadership and guidance steered this plenary towards its objectives. Sir, your wisdom and unwavering commitment, as ever, have been the backbone of our efforts. We also extend our sincere thanks to our beloved and dynamic co-chairs, Dr. R.M. Sundaram and Dr. M.J. Baig, for their valuable insights, which have enriched our discussions and added immense value to this plenary. And we are also greatly indebted to all our senior colleagues and living legends of rice research like Dr. B. Mishra, Dr. B.N. Singh, Dr. V.P. Singh, Dr. Ravindra Babu, Dr. S.R. Das, and Dr. A.K. Singh. And sincere thanks all these legends are with us for 3 days continuously and enlightened us with their wisdom. Sincere thanks are due to all our dynamic cooperators who have come from different parts of the country for their active participation and thought-provoking interactions, which have been the cornerstone of this event's success. The knowledge exchanged here, I believe, will undoubtedly spark innovation and collaboration. I wish all the delegates a safe journey back home and a happy reunion with your family. We express our deep sense of gratitude and sincere appreciation to all our generous sponsors whose trust and resources were pivotal in making this event a resounding success and also transforming our vision into reality. It is my duty to announce all the sponsors of this event. First, Fortune Rice Limited from Uttar Pradesh, who have sponsored the cake as well as the dinner. Pioneer Hybrid Seed Company, Bayer Life Sciences, Pan Seeds Private Limited, Harlal Seeds Private Limited, Advanta Enterprises, Savannah Seeds Limited, VNR Seeds Private Limited, Spring Bio Private Limited, Kaveri Seeds Company Limited, Indo-American Seeds, Rasi Seeds Private Limited, Seedworks International, DCM Shriram Limited, Star Agri Seeds, Lima Green India Private Limited, Coromandel International Limited, IFCO, Ambala Missionaries, Newsweed Seeds, and Shravan Scientifics. We profusely thank them for their generous contributions. Last but by no means least, a special tribute to our unsung heroes, the organizing team and volunteers whose meticulous planning and tireless efforts ensure seamless execution. We are deeply grateful for your dedication. And last 3 days, we are together with all of you delegates who have come all over from India and we enjoyed your company. And like all good things should come to an end, this event is also coming to an end today. And I wish you all again a very safe and happy journey back home. Thank you.
A special thanks to our veteran equipping leaders, particularly Dr. B. Misra, Dr. S.R. Das, Dr. Ravindra Babu, Dr. B.M. Singh, and Dr. B.P. Singh, Dr. A.K. Singh. They were last 3 days with us. They were guiding us. They were inspiring us. Thank you so much, sir. Thank you, your presence here. So please again get together next year for this program. Make it more inspired us so that we will be motivated. Thank you. Thank you all.
1 minute. So in commemoration of the Diamond Jubilee, we would shortly be organizing an international conference and the announcement will be coming soon. I would like all of you to participate actively and support. Yeah, in October end or something. Thank you all, and there is.
A proposal has come from Director CRRI Cuttack. He invites all of you to Cuttack. So next workshop tentatively, it is at CRRI Cuttack. It is tentatively. Of course, ICR will decide the venue at right time. Thank you. Thank you for your report.
Let us all rise for the national anthem.
अधिनायक जय हे भारत भाग्य विधाता पंजाब समुद्र गुजरात इंद्र हिमाचल यमुना गंगा उच्चल जगति तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे तव जय गाथा जय जय
Thank you, everybody. Safe journey home. There's tea served in outside. Please join us for tea.
With three AGRMs, I have photo with sir. Like this. Sir, shall I stand like this? हाँ? नहीं, ये आ जाएगा ना. Thank you. Thank you. Number I taken again. Again, no.